Piyush, are you there?
Yes, sir.
Piyush, can you run my slides once for me?
Okay.
पियुष
Yes, sir.
लोंढे सरांना परत लिंक पाठवा.
बरं सर पाठवतो.
काय त्यांचा प्रॉब्लेम आहे, त्यांना मिळत का नाहीये?
तर काही जणांना जात नाही झूम वरून मेल.
नाहीतर मी त्यांना शेअर करू का?
नाही मी करतो.
हा कर, आणि मिळाले का विचारून घे, कारण ही इज द फर्स्ट पर्सन टू स्टार्ट ना.
हो ओके सर.
ते आले की आपण कार्यक्रम सुरू करू शकतो.
वेदा सर.
हा या नेक्स्ट पियुष एकदा शेवटपर्यंत रन करून बघा, सगळ्या येतात ना. या परफेक्ट आहे पियुष, थँक्यू.
यू वेलकम.
सकाळी नेसलं जाते पाहुणे स्टेटस
Hello.
Hello. Good morning, Deepika sir.
Good morning. Good morning.
Morning. Can I just share the screen and see whether it's working fine?
Yeah.
Check.
Isha,
Yes, sir.
पीयूष ईशा को स्क्रीन पे ले लो.
Yes, sir.
You can start proceeding with Dr. Housie, sir.
Okay.
Dr.
Okay, sir. मग लोंढे सरांचं हे नाहीये का सर?
He will be joining later.
Okay, fine, sir. So कधी स्टार्ट करायचंय जस्ट सांगता. Start करू. Okay. Good morning, everyone. On behalf of the Pathologists Association, I, Dr. Isha Pansare, ex-committee member and honorary secretary, IMA Kalyan branch, welcome all the delegates and eminent speakers for today's webinar, Experts Conclave COVID Update Post Second Wave. Thank you, Dr. Dinkar Desai, sir, and team for giving me an opportunity to introduce our today's respectable guest. Our today's guest, Dr. Housie Kapadia. Sir is ex-state IMA MS president. Sir is family physician for 50 years. He is winner of several IMA MS and IMA headquarter awards. Sir has received Lifetime Achievement Award, General Practitioner Association, Greater Bombay. Welcome, sir. Now I request Dr. Dinkar Desai, sir, to take over for further inaugural proceedings.
Sir, please unmute.
Thank you. Thank you, Dr. Isha Pansare. Dr. Isha is presently Secretary of Kalyan IMA, and she was a founder member of TPA and ex-committee member. I welcome Dr. Housie Kapadia, sir, and I request him to do the proceedings of inauguration and address the gathering of TPA. Dr. Housie, sir.
Thank you very much. I thank Dr. Dinkar Desai and Dr. Isha Pansare and the organizers of the Pathologists Association of Maharashtra for this inviting me to grace this occasion of Experts Conclave, COVID update post second wave. There are a bevy of expert speakers who will speak on various topics. It will be an academic feast. The first and second wave have made around 1,500 doctors as COVID martyrs. We have learned a lot from where we were in March 2020. Now the prophylaxis, investigation and treatment are properly understood. Few drugs were rejected down the line. Vaccinations have also played a pivotal role in containing the disease. Right now, we are at the post-second COVID wave, and hopefully, if all prophylactic measures like mask, social distancing, and vaccination, if properly taken, we will be able to avoid a third wave. There is a lot of uncertainty about the third booster dose also, and they have started already giving it to some healthcare workers in the UK. It is most likely that we are heading towards a herd immunity. We will not be able to completely eradicate COVID, but it will affect us less complication and ICU treatment. It will be like any other flu virus affecting human beings. One more advice I would like to give to this learned audience, that please accept pathology reports signed only by qualified postgraduate pathologists in deciding the future line of medical or surgical line of treatment. I again thank the organizers for this calling me on this occasion to grace the occasion, and I am very happy that so many CMEs are conducted by the Pathologists Association. I again thank Dr. Dinkar Desai, Dr. Pansare. I wish the conference all the success, and here I inaugurate and open the conference for this further deliberations. Thank you, sir.
Thank you very much, sir. Dr. Isha.
Yes, sir.
Call Vanita. Dr. Vanita Korde.
Sir, yes. Am I audible?
You are audible.
Okay, sir.
Dr. Vanita
Good
Korde.
morning.
Yeah.
Yes, sir. Good morning, everyone. Thank you, Dinkar, sir. It's my pleasure to welcome Dr. Sonal Bangre, who would be speaking on everything about SARS-CoV-2. Dr. Sonal Bangre, an eminent medical microbiologist, is the founder and medical director of Infection Laboratories Private Limited, Mumbai. Infection Laboratories is the first and only specialized infectious diseases testing laboratory in India. Dr. Bangre has an experience of 16 years as an academician and an entrepreneur, providing services to the clinicians in the field of infectious disease testing, her areas of interest being syndromic panel testing, tuberculosis, and mycology. Dr. Bangre has completed MBBS from Grant Medical College and Sir J.J. Group of Hospitals, Mumbai, and MD Microbiology from Lokmanya Tilak Municipal Medical College, Sion, Mumbai. Apart from publications in national and international journals, she has been a keynote speaker in various national conferences. Now I would like to invite the chairpersons, Dr. Rupa Vishwanathan and Dr. Mukti Dave. Dr. Rupa Vishwanathan is the Chief Microbiologist and Managing Director of QualiLife Diagnostics and Joint Director of QualiLife Brain and Spine LLP. She has special interest in salmonellosis and tuberculosis. Dr. Mukti Dave is a microbiologist heading the Department of Microbiology and Molecular Biology in Infection Laboratories Private Limited, Thane. I now request Dr. Sonal to proceed with her presentation. Over to you, Dr. Sonal.
Thank you, Dr. Vanita, for a very elaborate introduction. I think I've been given the starting topic of introducing SARS-CoV-2, and it says all about SARS. So I'll just start off with my.
लौंडे सरकार ज्वाइन होणारे दुपारी कधी.
Okay, so this is a very interesting topic. Let's see a SARS-CoV through a microbiologist eye. So what my talk would be basically to create a background for all the upcoming talks which are coming in this day throughout, right? So let's understand the history of the SARS-CoV-2. Now, in 1965, there were two scientists, Tyrrell and Mayon, who found that they could passage a virus named B814 in the human embryonic tracheal organ from the respiratory tract with patients with common cold. At the same time, there were two other scientists called as Hamre and Prochnow, who were able to grow this virus with unusual properties from tissue cultures. Right, and these samples were obtained from the medical students with similar symptoms of cold. Now, both these B814 and 229 were ether sensitive and a lipid coat containing viruses, which were not related to any known of myxo or paramyxo viruses till then. So that is why this virus was named as OC. OC means organ culture, the virus which could be cultured only in the organs. So the earliest name of coronavirus was the OC virus. In late 60s, then these scientists named Almeida and Tyrrell performed an electron microscopy on the fluids from the organ cultures, which were infected with these B814 and 229 viruses. And they saw these particles. Now you can see these particles. This is the actual electron microscopic picture, where there were medium-sized viral particles which were pleomorphic, membrane-coated, and had those widely spaced club-shaped surface projections over it. And there were many other diseases that were studied or viruses that were studied during the same time. So they were studying some viruses which caused infectious bronchitis virus, mouse hepatitis virus, transmissible GI virus, in the swine, and all these, when studied under the electron microscope, had a very similar picture. So now these all group of new viruses were named as coronavirus. Now coronavirus, as the name denotes, it is the crown-like appearance of the surface projection, which was later officially declared as the new genius of the virus. So this is the actual electron microscopic picture, and this is the animation of the virus, how it would look like under an electron microscope. So when coronavirus was a very less known entity till a decade ago, and later on then it started gaining importance. Now, if you look at the genetic branching of the coronavirus, it comes under the family Coronaviridae, and it has two different lineages, two significant lineages. So it has alpha and beta coronaviruses, which causes infection in mammals, and avian coronaviruses, which are gamma and delta viruses. So there are two different lineages. What we are worried about, or the lineages which are undergoing mutations and coming up as new viruses, are the alpha and the beta. Of this, the major known coronaviruses were the human coronavirus 229E, the NL63, OC43, and HQU1. Now, these four viruses, usually caused mild respiratory symptoms all across. So most of the patients with mild symptoms used to get these coronaviruses. Now, are these coronaviruses new? No, they are being there since decades there, but it has a very less mutation which went. So the first four mutations were this. And then all of a sudden, in last 11 years, it started creating a new mutation series. Now, in those mutations were the SARS virus, the MERS virus, and the COVID virus as it's known today. Now, let's look at how this easy-looking microorganism, a virus, turned into a deadly virus trying to cause epidemics. Now, the first epidemic which came out was in 2003 of the SARS virus. Now, what is this SARS virus? Now, SARS is a coronavirus which is primary host is a bat. So this virus is coming from the bat origin, and it has taken civets as an intermediary host, and it had a little bit of a mutation there. So it is like the fifth mutation which happened in the coronavirus and which led to this big epidemic in 2013. Now, this SARS virus genome had around 87 to 92% sequence homology with the bat coronaviruses, and the spike protein, which was found to be very important. What is very startlingly different about the SARS virus is it has this spike protein which binds specifically to the ACE receptor. Now, the ACE receptors are there on the human lung epithelial cells. Now, the second mutation which happened and which led to another epidemic was in 2012, which was called as MERS virus. Now, okay, so MERS virus, and this was also transmitted through bats, which acted as the ancestral host. And here, the camels, they acted as the intermediary host. Now, again, it shared a genetic homology with the SARS-CoV-2 species worldwide. It was similar to the SARS virus, but only big difference between it was the receptor binding site. Instead of having ACE2 receptor, it had a DPP4 receptor there. Now, what is going ahead of this? Why is this virus is going mutation? Now, this COVID virus or SARS-CoV-2 is the 7th generation mutation. Now, why is there so much of a mutation in the SARS viruses? It is because of the major S1, S2 domain in the spike protein, which act as the major site of mutation, insertion, and deletion of genes during every transformation of the virus. Now, this enables the evolution of new infective virus forms that are capable of effectively binding the receptor domain sites, and hence increases the invasion. Now, let's look at the evolution and transmission of coronaviruses. Now, in the evolution, the bat plays a major role. Now, as you know, the bat species are the mammalian species which have a lot of viral infections. So, and there are 2, 3 major features about the bat because of which it is supposed to cause a notorious kind of viral infection. First is this multiple different species of bat inhabitate together. So you will find a fruit bat, some other kind of bats all staying in one place. So they have a transmission of viruses amongst them. Second thing is most of the bats travel long distances. So they can take one virus from one locality to another locality. Bats have a larger geographical area. So you can see those species all across the world. It is no country which doesn't have bats. And the third point is it has the ability to mutate because it is a mammal. It mimics the human viruses a lot. So most of the times what happens is bat provides as ancestral host, and directly from the bats the human being can get an infection. That is the main first chances of getting infection. Second is bats causes infection in the animals. Now these animals will act as a reservoir host. They will transmit infection between the animals also, and the human being coming in contact with these animals will catch in the infection of any virus. And the third, human to human transmission is very well known. It could be a contact infection or through infection through inhalation, through cough, cold, sneezing, surface contamination. Now, there are very interesting postulates of how excuse me, my PPT is not working. Yes, so there are many theories of origins of SARS-CoV-2. Now, this is being a very well-spoken topic all across. Now, how did the SARS-CoV-2 virus come out or COVID virus come around? So there are 3 postulates. The first postulate says that there is a natural selection in the animal host before the zoonotic transfer. What would that mean? That means the virus, as it is, the COVID virus got created inside an animal, and then it got transmitted directly to a human being. Now, this postulate, there are many things which are coming up to please pardon me, my PPT is not working very fine. Okay, so as many early cases of COVID-19 were linked to the Yunnan market in Wuhan. Now it is possible that the animal source was present in that location. That means there were bats which were sold in this market, and these bats had an infection which was very, very similar to SARS-CoV-2. And these bats act as a reservoir there. Second thing was these bats, when they were studied, they had their genetic, the coronaviruses that were found in these bats were almost 96% identical to the COVID virus. But what it had was it did not have that receptor binding site for ACE2 receptor. So this bat virus could not actually directly infect a human being because it doesn't have that receptor binding site. At the same time, in the same market, there were pangolins which were imported from the Malaysia. And these pangolins are supposed to have this RBD binding sites. So there was a postulate that between the pangolin and the bat, these coronaviruses got exchanged. And there was a virus which was created, which had this RBD binding site along with the COVID virus. Now, only thing missing in this, neither of this bat or pangolin beta coronaviruses had that polybasic cleavage. which could cause an infection in human being. And that is why this postulate may not hold very true, that there was a virus which was created already between the animals and it got transmitted to human being. So the second theory came as natural selection in humans following a zoonotic transfer. That means what? That the progenitor, the SARS-CoV-2, which was there in the bats, first got infected a human being. And it may have caused a minor infection in human being. Followed that, there was a human-to-human transfer. So let's take that picture of that same Yunnan market, wherein few of the vendors or the sellers of the meat got infection, and they had a small minor infection between themselves. And then during this infection, they acquired this human receptor, which is the ACE receptor. And this receptor, pangolins were also in the same market. So quite possible this infection went into pangolins, and then human to human transmission started. Now, there must be a small outbreak earlier in the market, and which went into creating an epidemic. Now, this postulate has a sufficient opportunity because many of these zoonotic events happen before the actual epidemic has happened. Now, the third postulate is selection during passage, that this viruses, basic research involving the SARS-CoV-2 is already going in the laboratories. So what is being done is they are trying to culture the SARS-CoV-2 viruses, not CoV-2, SARS virus, SARS coronaviruses, and there could be a laboratory release of this virus. It is also possible that they acquired this RBD mutation during a passage in the laboratories, but this cannot be anytime explained. So let's come at the morphologic or the genomic structure of SARS-CoV-2. Now, when it comes to a genomic structure, they have major structural proteins. Now, let's understand the structural proteins to understand how the laboratory diagnosis of these viruses happen. And it also helps you understand the development of vaccines and the drugs. Now, there are major proteins. There's a spike genome, which encodes for a spike protein. The spike protein is responsible for the virus entry. The S1 subunit will bind the host cell, and the S2 subunit will let it go through the cell membranes. There is a membrane protein which causes the virus fusion. There is a E antigen which causes the E protein. Then there is a nucleocapsid and a viral RNA. Now, if you look at the genomic structure, the major part, this RNA is the biggest RNA amongst most of the viruses. It is around 27 to 32 kb in size. It has a 5' guanosine cap and it is a poly A tail, which the genome, and the major part of it is a viral genome, which the testing. So ORF1 and B, 1A and 1B code for the proteins, non-structural proteins. And there is also a RNA dependent. The RNA also creates a polyprotein, which creates a RNA dependent RNA polymerase, which is RDRP gene. Now, these two genes are majorly used for the diagnosis of the testing of the COVID virus. Now, let's look at the most notorious part of this genome, which is a spike protein. Now, this spike protein is the one which is causing the mutations. So, as with other coronavirus, the entry of SARS-CoV-2 into the host cells is mediated by a transmembrane spike glycoprotein, which has it is a homotrimer. It is highly glycosylated, and it will be translationally cleavaged by a furin. Now, this furin is important because this furin cleavage, it is important for the infectivity and the virulence which is given to this virus. Now, the S1 subunit is going to bind with the ACE receptor, and the S2 subunit is responsible for the membrane fusion. Now, if you really see at the spike protein, this is the animation of spike protein that it continuously undergoes a conformational change between a closed conformation and an open conformation. So this circle here, it denotes the point where all the mutations are happening in the spike protein. So in this particular site, the spike protein keeps changing all the time. Now, what are the notable features of SARS genome? Now, the clean pictures which make it very different from the earlier SARS or the MERS virus is the mutation at the receptor binding site. So this ACE receptor binding site is undergoing mutation, and which is making it more pathogenic or more transmissible over the time. Basic furin cleavage, which was not seen in any other viruses earlier, is now seen, and there are O-linked glycans, which makes it quite resistant to immunoinvasion. Like other influenza virus or other viruses are, you can develop vaccines and they are very effective. But this virus was very difficult because of this O-linked glycan present in the SARS-CoV-2. Now let's look at the pathogenicity. So instead of me explaining this, I will connect to a video which very easily explains the pathogenicity of the virus. It will just take 2 minutes time.
There's no video visible.
Okay. Is it visible now?
Not yet, ma'am.
Okay, so I think I can skip it. So let's understand the pathogenicity of this. The virus.
Your slide is not seen right now.
Okay. Is it visible now?
Yes.
Hope the video is visible.
Yes, it is.
And this is the structure of the genome. The other spike protein. So, okay, so this was the pathogenicity, sorry, short video so that it could be visualized. Now, why do we need to know the pathogenesis of this virus? It is majorly to understand how the medicines will work. Where are the targets? So if you see, there is a spike protein binding site, and the drugs which will work on this site are these ivermectin. It is postulated that it worked at this site. The second was the ACE receptor site. The third was the TMPR site. Then the proteases, RDRP gene, and the endosome. So
Sorry,
chloroquine.
ma'am, please display your slides.
Sorry.
Your slides are not visible.
Okay. Hope it is visible now.
Yes, ma'am. Please continue.
So the pathogenicity needs to be understood to understand how the drugs, what would be the postulated drugs that will work. So as we understand, the virus first binds to the ACE receptors. So there were drugs which were used which would actually bind it would stop the binding of the virus to the ACE receptors. And this is where the earlier known medicines came into picture. So the chloroquine, azithromycin, those were first postulated because they would help stop the binding of these binding of the ACE receptor site. Now, there similarly, the other drugs were also postulated. Now, let's come at the mutations, because over the span of last, say, 1.5 years, there were multiple number of times when there are mutations which are coming up. Now, when you talk of mutations, it is a very common phenomenon in all the viruses. But what we would understand as mutation is the variation of concern. So when a mutation that will impact the virus characteristics, including its transmissibility or its antigenicity or its probable response to the changing immune profile of the human population. These are the features when we call it as a variation of concern. Now, there were mutations as early as around October 2020 from South Africa first. The major variant came up in December 2020, which was called as the UK variant, and then was a Brazilian variant and the recent Delta variants which came over. Now, there are some interesting facts about mutation in SARS-CoV-2. Now, why are there mutations coming in SARS? The virus that encodes the genome in RNA. Now, we saw that video wherein the genome of the virus goes inside the host cell, and it replicates there. And because of this, which happens in SARS virus or in HIV, influenza, they tend up to pick up mutations quickly as they're copied inside the host. And because the enzymes that copy the RNA are prone to make mistakes or errors. Now, after the SARS virus, the earlier SARS virus circulating, it also underwent a kind of mutation which causes deletion. And that is why all of a sudden the SARS virus, which the epidemic came in 2003, just went off, vanished. But now the sequencing data suggests that the coronaviruses change quite slowly. It is not actively mutating virus, because it has something called as a proofreading enzyme that corrects the fatal copying mistakes. So unlike the news articles are coming about the mutations being very haphazard, it is not so. It will only take around 1 or 2 single letter mutations, which is at half of the rate of mutation of about influenza or a quarter of a HIV virus. Now, despite of this very sluggish mutation rate, it has undergone around 12,000 mutations across in this 15 months time. But the scientist can spot mutations very fast. But to make sense out of that, it really takes time. You have to understand whether these mutations will have no consequences. So because they do not alter the shape of the protein or they do not create any significant change. Now, that is why there were only 4 major mutations which were found in coronavirus and which were variations of concern. So in those ones were the alpha variant. Now, alpha variant came around, say, December 2020, and it increases the transmissibility of the coronaviruses by 29%. So it increases the chances that the virus will spread. Now, this transmissibility is calculated depending upon the replication cycle of the virus. So the moment this virus, during this time, there was a lockdown put, the effect of this mutation got minimalized. There was increase in hospitalization with alpha variant up to 54%. Mortality increased drastically, almost like 59% for the age group more than 50 here. As far as the neutralizing antibody or the effect of vaccination was minimally reduced when it was a part of the alpha variant. Now, coming to the beta variant, which came in South Africa, it marginally again increased the transmissibility. Now, this increase in the transmissibility is in comparison to the original COVID-2 virus. There is not much data available from the South African variant because it did not spread much all across the globe. It remained somewhere stagnant into the African continent. It did reduce the response. I mean, the vaccination efficacy was reduced in the beta viruses and it still retained a severe disease. Now, very thankfully, the beta variant did not come into India much. The gamma variant, which was a Brazilian variant, again, it had an increase in the transmissibility and the mortality rates and a reduced response to the vaccination or natural infection. Now, the major variant of concern which came in India was the Delta variant, which originated in around October 2020, and we can call it as the reason for the second outbreak, the second wave, which came in April. So it increases the transmissibility by 97%. Now, that is a very drastic difference between the original coronavirus and the Delta variant. It increases the hospitalization by 85%. But when you compare it with the alpha variant, it increased by 147%. Now we can relate it to the second wave with all the hospitals getting clogged up and no oxygen available. It increases the mortality 137% in age group less than 50 also. So this was the concern which happened with the Delta variant. Reinfections happened after natural infection with Delta virus, but thankfully these infections are non-severe. So efficacy of the vaccination reduced, but even if a person gets infected with Delta variant post-vaccination, the disease is non-severe. Now let's see what happened in Maharashtra. Now, this was the alpha Delta variant which came up in Maharashtra, and it went around so many minor mutations, sublineages over the span of time. But neither of these sublineages went into a variation of concern. Now, you see the same spike protein here. It is the same point which is undergoing mutation again and again. It is so it is changing according to the way it wants to adapt to enter the human cell wall. Now, what is the impact of mutations? Now, scientists calculate the basic reproduction rate and evaluates the peak viral load and viral shedding time to understand the transmissibility. Now, what happens is when this spike protein mutates, it increases the transmission rate or reproduction rate of that virus. This can be easily controlled by lockdowns or social distancing. And that is why the need of social distancing is being over and over time postulated. It has an impact on diagnostics because these new variants may poses amplified risk of likely probe and primer mismatch. Now, what we are trying to test in the laboratories is the genes. Now you look for NRDRP, S gene, the ORF1A or 1B gene. These are the various genes which you are looking at. Now, if there are potential difference in one of these genes, some of the testing can go ineffective. This could obstruct the capability of the laboratories of present RT-PCR systems to diagnose. But till now, none of the variants have come such that it can totally escape the RT-PCR testing. Now, the third impact of mutation would be on the immune escape. To understand the affinity between antibodies and the variants, due to cross-reactivity of the variants of concern and elicited the neutralizing antibodies, the variant of concern can escape the immune system. Right, this can happen, and there could be impact in the vaccination. The development of next generation vaccine is one solution for vaccine escape. This should possess all the alternative epitopes other than from the different emerging variants. Till now, all the available vaccines are still relevant for all the variants of concern till now. Now, how are we to prepare for this? How are we going to look for this coronavirus henceforward? It is going to be there. It may remain in the system, in the environment, and we need to have a personal preparedness, a proper planning on the implementation stage. Policies which are, we have seen a drastic change in the Indian policy making in the last epidemic, and it is being very good. So this is all about coronavirus. I would say the king who wanted just more than the crown. It got the whole globe to a standstill. Just apart from being the major virus king, it has also got a lot of challenges to the human mankind. Thank you for your patient listening, because it was a very different kind of a talk. I would be open for any questions if there are any.
Thank you, Sonal, for that wonderful talk. And there are no questions in the chat box. I don't think I have any questions.
Yeah, it was a different topic altogether. We usually do not talk on basics so much. We would really like to talk on laboratory diagnosis and what are the new kits and what are the new methods that you could diagnose to coronaviruses.
So it was a very interesting talk, and I think already there are lots of, okay, there's one question. How will we come to know when the vaccines are not effective? So there's one question from Anupama.
Vaccines are not effective by laboratory diagnosis, because there is a coming talk on vaccine efficacy. So I think we should wait for those speakers to talk on knowing how the vaccine is not going to be effective. As far as laboratory diagnosis is concerned, there are changes in the S gene which are happening, which makes you understand there is a mutation which is happening. So testing with multiple genes is something that I would recommend, so that we do not miss out on the probable mutations that are coming.
There's one more question. Which vaccine is most effective for all variants? I think there is one. There is a topic on vaccination, isn't it?
Yes, there is a topic on vaccination.
So I think that question can be handled by that speaker. If you want to take it, yes, fine.
Yeah. Thank you, Dr. Rupa.
We'll wait for a few more seconds. There are no more questions as of now. So I think, Mukti, do you have any?
No, it was a very, very in-depth taken lecture. Yeah, we had a good brief about SARS-CoV-2, starting from maybe the history to maybe where we are heading towards. And yeah, no questions from my side too. Yeah, thank you.
Okay. So thank you, Sonal, for this lovely lecture. And I think it's like, it it's covered all the basics related to SARS-CoV-2, SARS-CoV per se, and SARS-CoV-2 as well. So I hand over to the organizers to take it further. Thank you.
Thank you.
Thank you, Dr. Sonal, for the wonderful session. Thank you, Dr. Rupa and Dr. Mukti, for chairing the session. Now I call upon Dr. Bhagyashree Ahir Rao to begin the next session.
Thank you, Dr. Vanita. After this elaborative session by Dr. Sonal ma'am, we are moving to our next session, that is by Dr. Jyoti Chidgupkar ma'am. Dr. Jyoti ma'am is a Vice President, Indian Medical Association, Maharashtra State. She is Vice President of TPA. She is an eminent microbiologist and in charge of government COVID lab in Solapur. I welcome you, ma'am. We are looking forward to your very important and practical topic, that is role of RT-PCR, rapid antigen test and COVID antibody testing. To chair this session, I invite Dr. Ninad Shah sir and Dr. Swaroop Kulkarni sir. Dr. Ninad Shah is a private practicing pathologist from Borivali. He is Secretary of Association of Practicing Pathologists India. He has interest in hematology and biochemistry and great knowledge of application of new technology in our pathology in day-to-day life. He has done his UG from GS SET Medical College, Mumbai. He has done his MD Pathology from Krishna Institute of Medical Sciences, Karad. Dr. Swaroop Kulkarni sir, presently Treasurer of TPA. He started Excel Laboratory in 1999 as the Thane's first immunoassay laboratory. Presently, he is lecturer at MGM Medical College, Navi Mumbai. He has done his UG and PG from Krishna Institute of Medical Science, Karad. I welcome both the chairperson and we will start the session. Hand over to Dr. Jyoti Chidgupkar ma'am.
Good morning, everyone. At the outset, am I audible?
Yes, ma'am, you are audible.
Thank you very much. At the outset, I would like to thank TPA and Dr. Desai sir for giving me this chance on this very esteemed platform of TPA. Thank you very much. To start with, am I able to show you my screen? Hello?
No, ma'am, your
can't hear
screen
you.
is not visible, no?
Now?
Yes. Yes. Yes.
Should I start now? Okay.
Yeah,
so about diagnostic testing of COVID-19 and RT-PCR antibodies and challenges all about this, I would like to highlight today. We all know that testing and contact tracing are essential for controlling the COVID-19 pandemic, as just now we heard in the very first lecture about the coronavirus, COVID-19 and pandemic. As many countries face ongoing waves of infections, demand for high quality diagnostic tests keep on growing, and at this moment we are again on the threat of third wave. So we should definitely go ahead with diagnostic new and new emergent techniques and yes. So already we have learned about heard about this coronavirus, but just brush up. We know that coronavirus is having spike protein, HE proteins, then envelope proteins, genome of RNA. It is plus ssRNA virus, nucleoprotein, and membrane proteins. SARS-CoV, which is from family Coronaviridae, is a beta coronavirus, and mostly nowadays we know all sorts of viruses, alpha, beta. We have seen in is an envelope positive sense single stranded RNA virus. Just I am briefing this to know further what RT-PCR is. The SARS-CoV-2 genome encodes 16 non-structural proteins, including RNA-dependent RNA polymerase, famously named as RDRP, and the helicase, NSP13, and 4 structural proteins, that is spike, membrane, nucleocapsid, and glycoproteins and envelope vaccines. So we have seen this figure also, where we have seen that the whole genome sequence of SARS-CoV-2 is about 20,903 BP in length, containing the following functional elements. These are poly A cap, 50 bases, the open that is ORF1 AB, containing coding sequences of RNA-dependent RNA IDRP, spike proteins, envelope proteins, membrane and nucleoproteins. I am again and again telling you just to know the basics of PCRs. S protein, a class 1 fusion protein, is pivotal for endocytosis. As we all know, endocytosis mediated viral entry, and consists of 2 subunits, S1 and S2. S1 harbors the receptor binding domain, that is RBD, which directly binds with the human angiotensin converting enzyme S2. Crucially, while the M glycoprotein is the most abundant SARS-CoV-2 protein, the S glycoprotein is the main inducer of neutralizing antibodies and the most diverging protein with a high mutation rate, possibly modifying glycosylation sites and consequently altering ACE2 binding, CTL epitope, and accessibility to protease and neutralizing antibodies, which may be important in mutations and resistance, all these things. So despite all these inventions and all these work in the COVID-19, the third wave, and in spite of having so many vaccines in pipeline as well as in working already in India, we have almost 50 crores of first vaccinations. People have been vaccinated. Still, there is impending threat of third wave. Third wave has been already experienced by UK, US, and other few of the other countries, and India is also on the threat. Maybe we are predicting that in November or December there may be a third wave. So in this context, we have to have more and more efforts to diagnose the COVID-19, and the work should continue. In addition to this, nowadays we have seen a lot of flu cases we are coming across, and also dengue cases and chikungunya cases, other viral fevers. So in addition, a great concern is also represented by the need to differentiate COVID-19 cases from seasonal flu that could clog hospital emergency services, slowing down the diagnostic and therapeutic procedures for the patient with COVID-19. So we have to take care of this also, and that's why in the case of COVID-19 pandemic, the effective test should be rapid, repeatable, based on technologies available at numerous centers, and keep costs limited in order to be carried out on a large fraction of the population. And population screening strategies should be available to screen for epidemiological purpose as well as immunization purpose. So this is a flow of, in general, a flow chart for the diagnosis. When patient come with symptoms, it is very important to have to do the test at right time, at right sample. So RT-PCR is first, or antigen is also advised. If it is negative, we have to go for judge the clinical probability. Then if still it is not high, if it is clinical probability is high, we have to go for serology, CT chest, and then we have to diagnose come to diagnosis. It is very mandatory to come to diagnosis. These are the overview of the available clinical diagnostics and diagnostic and research strategies for the effective diagnosis of COVID-19. In initial days, we all know that we were not having anything in our hands. So firstly, when the patients, lot of patients we started coming across such patients, clinicians, the clinical examination was only the test. Then gradually radiology also helped a lot to us, especially X-ray, CXR and CT scans. Then scientists started researching on this, and viral cultures came into picture, where Vero cells were used, and also next generation sequencing was tried. And then with the help of next generation sequencing and the sequencing methods, lot of tests came into the picture, and the common one were rapid antigen antibody testing. Then most popular test is RT-PCR based molecular test, and also serological test. Scientists also tried so many tests, and they are also in pipeline, and they are trying to get into the clinic, into the practice, all these tests like isothermal amplification testing, electron microscopes based methods. Of course, this is cannot be used routinely. Then biosensors, then CRISPR-Cas test, and digital PCR. So these are the firstly used tests. Next, viral culture, next generation NHG, clinical. This is repetition. These are the approved diagnostic methods, which we will see one by one. So diagnostic methods for this infection based on two parts. That is, one, direct identification of viral RNA. That's nucleic acid amplification test or antigen, or the indirect identification of specific antibody responses. A direct diagnosis is the gold standard for an active infection. So nucleic acid amplification tests are very important, and they play a pivotal role in diagnosis of COVID. While the detection of specific anti-SARS-CoV-2 antibody, also known as serological test, is cornerstone for the identification of previous contacts with the virus, both for diagnostic and epidemiological purpose. So this is a rough sketch of what sorts of tests they are available. Nucleic acid analysis detection by RT-PCR or RT-LAMP, antigen test either by ELISA or lateral flow, antibody test by lateral flow or again ELISA. So amongst these lateral flow tests are very fast. Within 15 to 20 minutes, we are getting the results. Though the sensitivity and specificity is low, while RT-PCR test and are taking 3 to 4 hours, the LAMP test is very fast. Within 1 hour, we are getting. This is a comparative chart for RT-PCR, RT-LAMP, and antigen testing by ELISA. Of course, all three testing we can do on nasopharyngeal or oropharyngeal swabs, or also lower respiratory specimens have been used. The sensitivity and specificity, of course, varies. Amongst these, RT-PCR and LAMP is very sensitive, almost 90% sensitivity is seen, and antigen test is just 70 to 85%. Again, this is a comparative chart where the same has been shown. Only thing is that the cost of antigen testing is quite low in especially lateral flow test, immunochromatographic test, but and otherwise the same sample can be used, and we can detect antigens as well as RNA. So who should be tested? Now, people who have symptoms of COVID-19. Then most people who had close contact with someone with confirmed COVID. Then even if fully vaccinated, nowadays it has been seen that Delta virus is becoming coming in existence and is settling. So it is necessary to go for COVID testing. But otherwise, it was said that fully vaccinated people with no COVID symptoms do not need to go for testing, and people who have tested positive for COVID-19 within the past 3 months and recovered do not need. But nowadays, just because of Delta virus and reinfections also, we need to test them. People who have taken part in activities, common activities, and have big exposure such as travel, social or mass gatherings, or they have poor ventilated indoor settings, they have gone into the crowded areas, they have to be tested to screen them. People who have been asked for or referred to get tested by healthcare providers is also important. They should have they should get tested definitely. So these are the different samples: anterior nares, then mid-turbinate, nasopharyngeal, and oropharyngeal samples. Amongst these nasopharyngeal, oropharyngeals, yield the virus yield from these two samples is great, and so preferred is nasopharyngeal and oropharyngeal. Though we have seen that people which they don't have good training, they collect the sample just from anterior nares. Sometimes they just touch the tip of the nose and say that yes, we have collected the sample. I have seen heard such complaints during screening. So it is very necessary to collect the correct sample and proper sample. So nasopharyngeal and oropharyngeal is advised sometimes for screening purpose, and especially after long time after exposure, a few samples like feces and urine sample also have been tried, and lower respiratory samples definitely give good yield, but bowel collection is not that easy every time. So nasopharyngeal and oropharyngeal samples are most preferred. So firstly, antigen-based test can be ELISA or can be lateral flow test, where we direct methods for these are the direct methods for COVID-19 diagnosis. Also encompass a double antibody sandwich ELISA, either direct ELISA or antibody sandwich ELISA also available, using nucleoprotein SARS nucleoprotein, and which is pre-coated at the base of the veil, and this can be used for the test. The direct method is simple, rapid, and does not require trained personnel and expensive laboratory instruments. However, in a meta-analysis, it has been seen that these are sensitive only for 70 to 86%, but specificity is definitely good. It is 95 to 97%, and thus a single negative test result cannot rule out the SARS-CoV-2 infection. We have to remember this. These are the lateral flow figures of lateral flow for antigen as well as antibody testing, where the antigen or antibody has been embedded into the chromatographic paper, and we are very routinely doing this test, which is rapid, cheap, but less sensitive. We all know about antigen. That test is very important and can use many different methods to amplify nucleic acid and detect the virus, including but not limited to. The very important test, which is very routinely done, is reverse transcript test, polymerase chain reaction, RT-PCR, and other tests, especially in researchers they have been used. These are the certain other isothermal amplification technique, including nicking endonuclease amplification reaction, transcription mediated amplification, loop mediated isothermal amplification, helicase dependent amplification, clustered regularly interspaced short palindromic repeats, CRISPR nowadays being famous, strand displacement amplification. So this is a simple schematic diagram for how the RT-PCR is done. First, swab is collected, then it is transported into the viral transport medium to the laboratory. Then where it is subjected to extraction. The extraction procedure, we extract the RNA from coronavirus RNA from this sample, and then they are subjected to amplification, and on the PCR platform, they have been identified and analyzed in a graphical manner. So this is the another flow. We can see again the diagram of the nasopharyngeal swab or oropharyngeal, or both can be collected. Then viral transport medium, then extraction of RNA, viral RNA. Then it can be also called as transcription, because we cannot amplify viral RNA. So we have to transcribe them into the complementary DNA and then amplify. You can see the picture at various temperatures in real-time PCR platform, and then we get the graph like this, where we have to set the threshold for fluorescence, and where the graph is crossing, these are called as the CT values, the copies per reaction. These are the CT values. These CT values, they have been once famous, and they were asking, have been correlated with the viral load, but it is not true. And then the results of RT-PCR are available. So real-time PCR technology is the most used SARS-CoV-2 RNA detection. In several real-time PCR tests, the primers were designed against the envelope E and RNA-dependent RNA, that is RD-RP regions. The E region was used for first line screening, while RD-RP region was employed for confirmation. Other researchers developed one-step RT-PCR assay detect where ORF1b and N genes of the SARS-CoV-2 were used. The N gene assay was used for screening, while ORF1 testing served as a confirmatory test. However, since ORF1b and N regions are highly conserved, the specific primers could also bind other specific could bind also other coronavirus. So which are, and also closely related viruses. As regards the RT-PCR performances, a recent meta-analysis performed by Kim et al. Considered 19 studies, identified a pooled sensitivity of 89%, while positive predictive value it was 47 to 98%, and negative predictive value was 93 to 99%. In fact, false negative results may not only derive from poor sensitivity, but they can also be due to low amounts of samples, inadequate conservation, or presence of amplification inhibitors or mutants in the target region of the, or mutation in the target region of the specific primers. So we have to take care that the proper sample at right time is very important, and right amount of sample is also very important. Unless, though this is very sensitive, we may get false negative test also. In real-time PCR, the amount of gene target present in the sample typically determines the number of PCR cycles needed before SARS-CoV-2 is detected, and that is called as cycle threshold or CT values. All current SARS-CoV-2 RT-PCR assays with EUA approval are labeled only for the qualitative detection. We have to remember that they are not quantitative assays, and it is a qualitative detection for specific gene targets specific to virus, and not approved for quantitative measurement. While CT values can be reduced by increasing the amount of gene target in the sample, it can also be influenced by many other factors such as quality of the specimen, technique of the collection, sample type, whether it is nasopharyngeal or saliva or something like that, gene target, and type of the assay, There are no current recommendations for use of CT values in patient management, and more research on viral load kinetic is needed. So we don't have any quantitative as of today assays. An alternative to fluorescent detection includes sequencing of the viral genome. We have heard about sequencing yesterday. While this has had limited applications in the detection of SARS-CoV-2, it can play an important role in determining whether a patient has become reinfected, as it may be possible to compare the sequence of original isolate with that of the second one. So this is again a schematic diagram. If patient is symptomatic, antigen is positive, he is infected. If asymptomatic and still antigen positive, we can confirm with a NAT testing. Then if asymptomatic and no exposure, again antigen can be done, or RT-PCR is the important test for such patients. Indirect diagnosis. Now I'm going towards the indirect diagnosis or point of care test. We can do the indirect diagnosis of SARS-CoV-2 infection is based on the detection of specific antibodies, that is IgG or IgM. Importantly, the two underline WHO underlines strong need for serological IgG IgM testing and rapid design of such tests, which are inexpensive and can be used as point of care tests, just like antigen testing in lateral flow. These are the just we can have antibody testing at the used, and ELISA also can be used for that. This is the schematic diagram for ELISA-based immune enzymatic serological test. Analytical workflow of indirect ELISA for the effective detection of IgA. IgA being early protein can be detected, can be used for, nowadays they are trying for early detection of COVID. IgA, IgG or IgM, and analytical workflow of sandwich ELISA also mentioned here, where directly antigen is embedded at the web base of the well, or antibody complex is embedded. So this is the sandwich and direct ELISA. Now exactly what time determines or what test should be done should be determined by the time or period of the disease. So we know that up to 7 to 8 days there is window period where patient is asymptomatic, and then onset of symptoms starts with the detection. So antibody mediated immunity in SARS-CoV-2 virus specific IgG and IgM are detectable in serum between 8 to 14 days. Though IgM is early protein, you can see IgM is early protein. It starts rising early and it drops early by 21 days. While IgG is remains there in blood for longer time. Maybe up to 7 months, they say it can be there. Viral RNA is inversely correlated with neutralizing antibody titers. As neutralizing antibody titers rise, viral RNA drop. Higher titers have been observed in critically ill patients, but it is unknown whether antibody response somehow contribute to pulmonary pathology. The SARS-CoV-1 humoral response is relatively short lived, and memory B cells may disappear altogether, suggesting that the immunity with SARS-CoV-2 may wane 1 to 2 years after primary infection. So we may predict that there may be requirement of booster dose after 1 year, which is under discussions. So after during window period, we may detect RNA or antigen in late window periods especially, and IgM earlier, IgG, IgM being early immunoglobulin, IgG will be detected later. And again, nowadays we all know that IgG against nucleoprotein and against spike protein are helping us to know whether cocktail should be given to the patient or no. after vaccination, especially. Serological testing does not uh, uh, it is not a diagnostic, but should not be used to establish the presence of or absence of acute SARS-CoV-2 infection. Antibody testing not currently recommended to assess for the immunity to COVID-19 following COVID-19 vaccination, but we can know that whether antibody response is there or no, or to assess the need of vaccination in the unvaccinated persons. Since vaccines induce antibodies to specific viral protein target, post-vaccination serologic test results will be negative in person without history of previous natural infection if the test used does not detect antibodies induced by the vaccine. So vaccination, again, vaccine induced antibody developed by has implications for serological testing. Before vaccine introduction, a SARS-CoV-2 serology test that detect any of the S, N or RBD antibodies could be considered to indicate previous exposure to SARS-CoV-2. But now after vaccination, it is not possible. Thus, history of vaccination and or prior SARS-CoV-2 infection must be considered when interpreting serological test results. So there are certain do's and don'ts until scientists get more data on how much protection antibodies provide against being infected again with this virus. Everyone should continue to take steps to protect themselves and others, including wearing masks, staying away, not going into the crowds, and washing hands, even if they have had a positive antibody test. People who wear personal protective equipment at work should continue to wear PPE, even if they test positive for antibodies to virus. What are the don'ts? Antibody test results should not be used to determine if someone can return to work. Antibody test results should not be used to group people together in settings such as schools, dormitories, or other facilities. It just vaccination, to have vaccination is important. To get vaccinated is important. So these are the certain antibody tests like CLIA, ELISA, or immunofluorescence we have used amongst these. Again, the same, these tests, they can be used commonly of these ELISA, CLIA and fluorescent antibodies. Fluorescent tests, they have been proved more useful and fast as compared to ELISA. Now research methods, nowadays certain research methods, they are available and they are being evolved, and they have been tried to use in diagnostics also, like isothermal technique, electron microscope methods, biosensors, CRISPR-Cas based diagnostic methods, and digital PCR. This is a schematic flow of nucleic acid isothermal amplification method for the diagnosis of COVID-19, where RNA extraction and then RT-LAMP is technology is used. These are certain alternative methods. LAMP may come into the picture soon. It looks like that. This is CRISPR-Cas based technology for diagnosis of COVID-19. CRISPR-Cas systems effectively detect SARS-CoV-2 viral RNA after complementary DNA synthesis through the recognition of specific SARS-CoV-2 sequence that is cleaved by Cas12a activity, resulting in the collateral cleavage of fluorescent probes, and even Cas13a activity also used, and then immunoassay. This CRISPR is cluster of regular interspace short palindromic repeats Cas, and then with the help of fluorescence we can detect the RNAs. This is digital droplet digital PCR. Again, new technology is being evolved where again viral RNA are retro-transcript to complementary DNA amplified, and with on this platform So many of the platforms they are available for this digital PCR. We are able to detect the viral RNA. This is just a photograph of mucormycosis, which became rampant after especially after second wave, where the diagnosis initially was quite confused, but later we could do and we could conquer mucormycosis also. And commonly used methods are microscopic methods, serological methods, and certain molecular assays are also available. This is diagnosis we can see in a HE staining. This hyphae of mucor, and this is colonies of mucor, and microscopic picture of the mucor. So in the impending a scenario of third wave, we have to think of diagnosis. We have to continue doing and continue the researches in the diagnosis of COVID-19 and get more and more easy tests and less expensive tests, and try to kick out the COVID. Already we have done a lot of work on COVID and tried to kick out it. But now this before coming of the third wave after vaccination, we have to kick out this COVID coronaviruses and get free of this pandemic. Hope for earlier declaration of finishing of the pandemic. So thank you very much for patient listening. Thank you very much. Over to you, moderators. Thank you very much.
Thank you, madam. Very simple and easy to understand presentation. We have a few questions regarding method of testing. Starting from if a patient cannot tolerate some nasopharyngeal swabs, what other methods of collection do you suggest which can be used in majority of the RT-PCRs or LAMP test or say RAT test?
Actually, nasopharyngeal and oropharyngeal yield is maximum. It is highly sensitive samples, and for the testing, it is very important, as I said, to get the correct samples. Correct sampling is very, very important. As we collect the samples from nares or turbinate, the sensitivity of the test decreases. So we have to have those samples, but saliva also can be tried, which is very easy. For bedridden patients, we can collect it or if they are on endotracheal intubation, we can collect those secretions also, or even sput-up can be, and bowel fluids can be tried for this.
Ma'am, normal saline gargles can be used?
We can use, even saliva can also.
Better.
Yes, saliva is better. Saliva is better.
Next question regarding antibodies. There are a lot of queries that post-vaccination, how is my immunity? So sometimes we see nucleic acid antibodies against Covishield. For other vaccinations, how do we conclude, say for Sputnik or for this one, Covaxin? So any chart, flow chart or something, if possible, whether it is post-infectional or this thing?
Actually, I could not get such chart, but I can tell you after infection, as we know, IgG and IgM and IgG will be detected. Amongst it, IgG, which can be against either nucleocapsid protein or from against spike protein, because those which can have some protection, or those which have good immunity, they can produce against spike proteins, IgG spike proteins. But especially the more high amount of IgG antibodies against nucleocapsid are after infections. Whatever I have seen doing these tests, that in early stage we get IgM protein positivity after infection, then IgG, which is mostly against nucleocapsid, or total. We have commercially available tests which are against total antibodies. So we can get a high number of total IgG antibodies. But after vaccination, we have seen that the antibodies to spike protein, because as we know, after antibodies, vaccines should stimulate the production of antibodies against spike proteins, which are protective in nature. So after vaccination, the amount of vaccine antibodies produced against spike proteins are quite high as compared to nucleocapsid. Any comments from Bhagyashree, ma'am?
Hello. Yes, I was also wanted to know the same happened because after the post-vaccination period, many doctors themselves asked to test the antibodies.
Yes.
So that was the scenario. But I think after your lecture, most of we got the idea that it's not necessary to do it.
Yes, because
So,
as we all know that T cells are also stimulated, and it is very much known for viral infections that T cell stimulation, have pivotal roles. Cellular immunity plays a pivotal role in protection, and that's why we get many a times lifelong protections against certain viral infections. So actually, this is again under research. I think we'll know gradually how much protection by cellular immunity we are getting. But as of today, only key to know about the post-vaccination immunity is antibody testing, which I don't think it will these antibodies will be lasting after 8 or 9 months in blood.
and I just want to add, ma'am, because there was also a letter from Government of India not to do antibodies after vaccination.
Yes, absolutely. Yes, Dr. Nina Shah, any other questions?
Thank you, madam. Thank you for a great lecture. What I basically wanted to know that if there is a discrepancy between RT-PCR testing, what to do? Because sometimes we are in a dilemma because our patients are hyper and they do test from multiple labs, and one of them may be negative and other may be positive. We understand the sample taking is a major thing which most of technicians don't follow standard protocols. So there is pre-analytical bias already present. But in such scenarios, what do you recommend? We should take up that positive to be true positive, or there might be false reactions.
See, sometimes even if patient asymptomatic or symptomatic or having strong history of contact, or even suggestive symptoms, suggestive radiological pictures, and we are getting RT-PCR test negative, we can go for repeat testing. For 3 consecutive days, we can go for repeat testing and find out whether it is positive or no, because sometimes window period may hamper the diagnosis of the the laboratory diagnosis. And in case of discrepancy, of course, see, antigen testing, we know that they are not that sensitive. So sometimes, and RT-PCR is always confirmatory test. We have to go for RT-PCR, and we can repeat it definitely in case of discrepancy.
Is there any role of post testing after 14 days, or it is not recommended?
Actually, not recommended. It is not recommended. After 14 days, patient is said to be negative if patient is not having any more symptoms.
Swaroop, to you. Over to you.
Dr. Ninat.
Any other questions from the house? I think so. There are no more questions.
Dr. Swaroop sir, any questions from your side?
No. I think most of the things have been covered very well by you, and some queries that were there were answered also right now. So
Dr. Swaroop, can I add something? I wanted to.
yeah.
Sure, please.
Dr. Swaroop, only one thing I want to add. Like Dr. Nina told that some lab it is giving positive, some lab it is giving negative. In that case, we have to counsel the patient in the way that that single positive test is positive. Patient will ask you whether I'm positive or negative. So if some lab is giving positive, we have to consider that patient as positive. First thing. And second important thing, I have second important question which was asked during April to July, most frequently by general practitioners and physicians, which we couldn't answer, which I want to ask Madam, that can we differentiate Delta variant from the rest of viruses? Do you have that kind of primer where you can do, or is it going to be available in future? Just
I
telling
think
that
they'll
it is
get
COVID positive.
as of today, I don't think any differentiation in by RT-PCR testing between these variants is possible. But only thing they may add the primers and probes, but sequencing is needed for this. And may the sequencing be available in near future in laboratories, our diagnostic laboratories also.
Okay, madam. Thank you. Thank you, Dr. Nina, Dr. Swaroop.
Thank you very much. Thanks for giving me the chance. Thank you very much.
Thank you, Dr. Jyoti Chirgupkar ma'am. After this lot of questions, we understand this was the most important topic for all of us. I also thank chairperson of this session, Dr. Ninad Shah and Dr. Swaroop Kulkarni sir. Now for the next proceeding, I will hand over to Dr. Janvi Ahuja. Thank you.
Everyone. So are we inviting Dr. R.S. Londe for the 5-minute address? Okay. So I welcome Dr. Dinkar Desai sir for the next session on lab monitoring in COVID-19 patients. Dr. Dinkar Desai sir is the president of the Pathologist Association. He is the former vice president of APPI and IMA Maharashtra State. He is a practicing pathologist at Thane and holds triple degrees in medicine, law, and management. Welcome, sir. May I also invite the chairperson, Dr. Munjal Shah. Dr. Munjal Shah is MBBS from Grand Medical College and Sir JJ Group of Hospitals, Mumbai. He is MD in Pathology from Tata Memorial Hospital, Mumbai. Currently practicing pathologist and partner at Paras Pathology LLP, Santa Cruz, Mumbai. He has keen interest in academics, research, and teaching. He has been a visiting faculty at technical colleges. He is active in writing medical articles as blogs for patient education on the website paraspathology.com and has revised and rewritten PJ Mehta's Practical Pathology. I also invite our chairperson, Dr. Rajesh Butala sir, who is an MD and DNB in pathology, running a diagnostic center by the name Lifeline Diagnostics for the past 18 years. He is the secretary of the Pathologist Association. Thank you. Sir, you are muted, sir. Desai, sir, you're muted.
Sorry. So, thank you, Dr. Janhavi Ahuja, for giving the best introduction. My topic of today's lecture is lab monitoring in COVID-19 patients. We have got brief knowledge about the virus and what tests, laboratory tests, especially of microbiology, we are performing like RT-PCR antibody tests. Pardon? Yeah. We just saw how the strain differs from the first wave in the second wave and what were the mutations. Silent observations like age pattern, bypassing the RT-PCR by the mutant organism, and more thrombotic complications noted in second wave. So these things we have just now studied, so I will not go into the details of that. Lab tests for management and monitoring. So where we are interested in the monitoring and management of the case which is most probably admitted in the wards or ICU. CBC, NLR, RDW, eosinophilia and lymphopenia, C-reactive protein, ferritin, interleukin, IL-6, D-dimer, LDH, PCT, procalcitonin, arterial blood gas analysis, liver function tests like SGOT, SGPT, S-bilirubin, creatinine. First, we look after the NLR, that is, pardon? Dr. Mavarkar, please unmute yourself. Yeah. NLR, that is neutrophil to lymphocyte ratio. It is the ratio of absolute neutrophil count to absolute lymphocyte count. Remember the word absolute neutrophil count. Virus triggered inflammation increases the NLR. Elevated NLR levels more than 3.5 is an independent prognostic biomarker that indicates infection severity. NLR elevation may be due to dysregulated expression of inflammatory cytokines or an aberrant increase of pathological low density neutrophil or the upregulation of genes involved in the lymphocyte cell death pathway. NLR has the highest of sensitivity and specificity and is a very superior marker to other inflammation-based prognostic cases. Next is RDW. An elevated RDW, that is RBC distribution width of more than 14.5 is a very simple marker of progression and is associated with an increased risk of all causes of mortality. The causes of mortality like pulmonary disease, sepsis, influenza, and cancer, complications associated with heart failure and viral hepatitis, advanced stage and grade for many cancers, and the development of diabetes, chronic obstructive pulmonary liver disease, stroke, anemia, and many other conditions. An increase in RDW during admission or over the period of hospitalization was associated with increased mortality. Now look at eosinophilia. Absolute eosinophil count less than 10 per cmm has been reported in coronavirus outbreak and is characteristic of COVID-19 pneumonia. A zero eosinophilic syndrome indicates a very bad prognosis. Similarly, lymphopenia is found in 80% of the critically ill patients. C-reactive protein, normal range up to 8 mg/cent. Measurement of C-reactive protein is useful for the detection and evaluation of infection, tissue injury, inflammatory disorders, and associated diseases. As per some studies, higher the C-reactive protein, use higher the steroid dose. That is the dictum as far as the monitoring and management is concerned. It was observed that patients with low oxygen saturation, that is SpO2 less than 90%, had significantly higher levels of C-reactive protein compared with patients with high oxygen saturation, that is SpO2 more than 90%. Combination of eosinopenia and elevated C-reactive protein can effectively triage suspected COVID-19 patients from other patients attending the fever clinic. So this is a major problem faced by the hospitals who are non-COVID. They have got their triage ward, so they don't know whether this patient is coming with in fever clinic with fever, is going to be admitted in their own hospital or is to be shifted in COVID hospital. So when the patient is in a triage, a combination of eosinopenia and elevated C-reactive protein gives a better indication for what you can say to shift him either way. Ferritin. Oh, before that, C-reactive protein. Measurement of C-reactive protein is useful for the detection and evaluation of infection, tissue injury, inflammatory disorders, and associated diseases. Oh, I want to come to ferritin. Normal range is male 20 to 500 nanograms per ml. Ferritin has been characterized as an acute phase reactant as well as a mediator of immune dysregulation in severe COVID-19. So is an active player in the cytokine storm scenario that characterizes severe COVID-19. Ferritin is associated with the severity of lung involvement, but not with the worst prognosis in patients with COVID-19 disease. Interleukin 6 normal range is 0 to 7 picogram per ml. IL-6 is the most robust indicator or predictor of hypoxemia. The concentration of IL-6, if it is greater than 24 pg per ml, is predicted the development of hypoxemia with the sensitivity of 100% and specificity of 88.9%. So very important test, interleukin 6 in case of severe cases of COVID. Sequential measurements of IL-6 in ICU patients is useful in evaluating systemic inflammatory response syndrome, sepsis, and septic shock. Increased levels imply cytokine-mediated lung damage in patients with respiratory dysfunction. Cytokines are vital in regulating immunological and inflammatory response. But interleukin 6 has got a curse. Many times you find pre-analytical errors and very high levels of interleukin 6 are found in cases of normal patients without any COVID severeness. These pre-analytical errors, IL-6 values are spuriously high in these errors due to improper blood collection tubes, plain tube. The preferred tube is EDTA. There are transport delays and transport and temperature exposure that has to be avoided. A very old test now has come in focus, and that is the D-dimer test. D-dimer is the product of fibrinolytic degradation of fibrin, and elevated levels indicate that there is a hypercoagulable state and secondary fibrinolysis in the body, which is extremely useful for the diagnosis of thrombotic diseases. When a clot is breaking down, D-dimer hangs out. D-dimer is so named as it contains 2D fragments of fibrin joined by a cross-link. If D-dimer is greater than 1 microgram per ml, suspect deep vein thrombosis or pulmonary embolism, DVT or PE. It's a wonderful prognostic marker for monitoring hospitalized patient, and that is the reason why it has got such a lot of importance in the COVID times. D-dimer has a negative predictive value. This is most important. It has got a negative predictive value of 100%. Normal D-dimer, it means there are no clots. LDH is one more test which is frequently asked. The normal range, 100 to 250 units per liter. LDH is an enzyme implicated in the conversion of lactate to pyruvate in the cells of most body tissues and increase following tissue breakdown. It's a non-specific indicator of cellular death in like many diseases such as cancer, liver diseases, sepsis, hemolysis. The most important thing is an increase in LDH by 62.5 units per liter has an acceptable sensitivity and high specificity for a disease progression probability. Means the values are higher than normal by 62.5 or more percentage, then you can definitely have got a case of COVID in your hand. Procalcitonin is the propeptide of calcitonin devoid of hormonal activity. Normally, it's produced in the C cells of thyroid. In healthy persons, PCT levels undetectable, less than 0.1 nanogram per ml. During severe infection, that is bacterial, parasitic, fungal, with systemic manifestations, PCTB levels may increase over 100 nanogram per ml. PCT in initial days is to rule out secondary co-infection and not to assess the severity of COVID. Most important. Not to assess the severity of COVID, but to rule out any secondary co-infection. Values are influenced by comorbid conditions like kidney disease, CKD, CCF, etc. Arterial blood gas test is useful in hospitals for measuring arterial oxygen and CO2 in severe cases. Monitoring has to be daily. Another test, troponin, to look after the cardiac injury, liver function test, to look after the liver injury, mild in moderate cases and moderate derangement in severe cases of COVID-19. Renal function tests are done to note the kidney injury. And this is the most important and last chart of my presentation. Lab findings in mild, moderate, severe cases. Nuclear lymphocyte ratio less than 3.5 in mild, it's greater than 3.5 in moderate, and more than 5.2 in severe cases. C-reactive protein in mild it is less than 20, moderate cases is 20 to 50, and in severe cases it is greater than 50. This is how the laboratory helps in the monitoring of mild, moderate and severe cases, and the clinicians can know which patient they are dealing and which the case is in mild stage or severe stage. Ferritin less than 500 in mild cases, moderate cases it is 500 more, and it's more than 800 units in severe cases. D-dimer, as you know, is less than 0.5 in mild cases, moderate in 0.5 to 1 in moderate cases, it's greater than 1 in severe cases. LDH less than 300, 300 to 400 values in moderate, more than 400 in severe cases. IL-6 less than 5 in mild, 5 to 50 moderate, and more than 50 or rising in severe cases. IL-6 has to be done in a sequential manner when the patient is admitted, and that reading has got more importance. LFT is normal in mild cases more or less. There is a very slight derangement in moderate cases and moderate derangement in severe cases. Piyush? Piyush?
Yes, sir.
Can you show the video for D-dimer?
Yes.
Yeah. It's a very interesting video I got. I just thought will be good for audience. Dr. Ramakrishna Londe sir is there in the audience, panelist. You are seen, but your audio and video is not on.
Sir, what was that file name?
That PowerPoint I said na.
Practical pathology.
YouTube का था.
YouTube पे था.
हाँ, T-Dimer.
No, sir, I don't have it at hand right now.
Huh?
I don't have it at hand right now.
Okay, it's okay, fine. Thank you. Thank you. Dr. Tanvi? I invite...
Thank you, sir, for the excellent presentation, very precise to the point. May we now have questions?
I think
Dr. Munjal and Dr. Butala, sir, any questions?
No, Janvi, we are getting very far late.
Okay, sir.
So half an hour we are late.
Okay, sir.
We can discuss afterwards. Dr. Munjal Shah?
Yes, sir.
पीयूष,
Yes, sir.
डॉक्टर मुंजल शाह को स्क्रीन पे लाना ना जरा ऊपर.
Piyush, you have put Dr. Mahavarkar.
Yeah.
Add Dr. Munjal Shah instead.
Good morning.
Yes.
Can you hear me, sir?
Yeah. Dr. Bondwe sir is there. Dr. Bondwe sir, can you put on your video audio? Phone kar Avinash Bondwe. Dr. Munjal, your video is on?
Yes, sir. My video is on.
Oh. Video on करा मला सर व्हिडिओ ऑन करा ना व्हिडिओ नाही करा लगेच घेतो. डॉक्टर मुंजल तुमचं पुस्तकाची तयारी आहे?
Yeah, sir.
Piyush, I am not finding Dr. Munjal. Yeah, now it is there. So, friends, Dr. Munjal Shah is one person. Piyush, Dr. Mahavarkaranच्या जागी तुम्ही डॉक्टर बोंडवे घ्या ना.
ओके.
आणि मुंजल व्हिडिओ ऑन करा.
हॅलो देसाई.
हा
काय कोणी माझ्या जागेवर?
नाही तुम्ही तुमचा काय प्रॉब्लेम नाही, आमच्या इथला इंटरनल प्रॉब्लेम.
ओके, नो प्रॉब्लेम. मी थांबतोय.
हा थांबतो. डॉक्टर बोंडवे सरांना घ्या.
डन सर.
कुठे मला दिसत नाहीत, पहिल्या याच्यामध्ये येऊ देत ना
सर. येते टॉप 4 मध्ये 4 आहेत
ते तुम्ही पिन केले त्याच्यामध्ये डॉक्टर महावरकर अनपिन करा आणि तिथे घ्या.
Sir, I can visualize you.
Yeah. Now it is there.
I'm there, sir.
I can see you.
I'm there.
Yeah. Sir, we welcome Dr. Avinash Bondwe, who is ex-president of, immediate past president of IMA Maharashtra State. And I am grateful that he has joined us. Dr. Munjal Shah, who is the chairperson of today's session, he is very young and bright. Along with his wife, Dr. Nehal, they are doing some lot of activities in pathology. And recently they have come up with a novel book on general pathology. So we thought our member has done a lot of good work in the field of literature, and it is the right occasion to release the book in front of our August gathering of pathologists. Dr. Munjal Shah also agreed, and his publication also gave. Are somebody present from their side?
No, sir. Actually, Vaidehi is there, the publisher. I think she is in the audience, I suppose.
So we wish them lot of thanks from TPA side and from Munjal Shah side. Mehta, na? PJ Mehta, someone.
Yeah. So Dr.
PJ
PJ
Mehta,
Mehta's practical pathology.
Yeah. So thank you, Doctor. Thank you, Mr. PJ Mehta and Popular Bookstall for releasing a book on pathology. We pathologists feel themselves that we are very neglected people of all the medical fraternity, and somebody is going to publish a book on pathology is very great incidence. Dr. Munjal, you would like to talk about your book? Hey, Piyush, can you show the highlights of the book?
Yes, sir.
Please.
I'll stop your sharing.
Yeah, stop my sharing.
So first and foremost, I would like to thank TPA and Dr. Desai and team for this encouraging event. This group has always encouraged young and newcomer pathologists like me who are at our infancy of practice to share the stage with the stalwarts, and nothing can be more rewarding than that. So that's my book, PJ Mehta's Practical Pathology. I have revised and rewritten the second edition with the motive of safeguarding the legacy of the great teacher, late Dr. PJ Mehta. We've all read at least one of his books during our medical journey. I suppose all of us can resound with that. When I was approached to revise this book for its second edition by the National Book Depot, it was a fan moment for me. So as a pathologist, I have added certain chapters like introduction to microscope, quality control, workflow in a lab, so that the medical student who is going to be the specialist of tomorrow knows how a medical laboratory works and what pains the pathologist takes to give a correct report. Going by the fast pace of technology, we've removed the pictures, which are easily available online, so that it reduces the cost of the book. And we've given numerous flowcharts and tables to inculcate the concept of algorithmic approach to each case, which helps keeps sanity in the vast ocean of pathological diagnosis. We hope that our efforts help the medical students in understanding pathology better. I once again thank TPA for giving me this opportunity to launch these books on this platform. Thank you very much, Dr. Desai and team. And thank you, TPA, Dr. Avinash Bondwe, for launching this book on this esteemed platform. Thank you so much, sir.
Sir, I would like to say something. May I?
Please. Yes, you're requested.
Yeah. Thank you, Dr. Dinkar Desai, sir, and organizers of today's webinar for inviting me in a very useful seminar. I personally congratulate Dr. Munjal Shah for his book on practical pathology, because all of you know that a doctor's, especially a doctor from modern medicine, our duty is not to treat patients, but to rather prepare our next generation and our colleagues with whatever knowledge we gain. As you all know, that most of us have tremendous data, tremendous experience, and extensive study and all these things. But these things are not passed on to the colleagues as well as for the students. The main reason for the fall of the Ayush medicine, especially the Ayurveda, is decided previously that the things were not passed on properly to the students. It was said, Dr. Munjal Shah, it was said that every guru of these Ayurvedic doctors. In past, they used to keep one jadi buti with him and then pass on to the students. What are the 99 jadi butis are passed on. So that was one of the reasons that nothing was documented, nothing was published, and whatever knowledge we have is not, rather they never wanted to spread it. So this is very good thing you have done, and I definitely look forward that in future also you will publish more and more books on different topics, and definitely it is a thing. And I rather appeal to all the people, all those who are attending this webinar, that keep on writing, because writing is very, very important. Personally, in my life, I have published around 13 books, and my motto is only to make people aware. As you all know, even the literate people, even the educated people in India are not health literate. Our literacy is going up, but the health literacy is not there. So it's our duty to spread this knowledge and to make people literate, to make our colleagues more and more knowledgeable. So thank you very much and wish you all the best.
Sir, may I add just one more line here?
Yes, sir.
Sir, as you rightly said, we should all be writing, and which as by the prefix of doctor, which actually means a teacher in Latin. So we should all be fulfilling that, and I'm really happy to share the stage with a person like you who's written so many books. That's an honor for me, and thank you so much, Dinkar sir and the TPA platform for this opportunity. Thank you.
Thank you.
Thank you.
Thank you, sir.
Thank you, Dr. Mondve sir. Thank you, Dr. Munjal Shah sir. Dr. Londe sir, can you put on your video? Mr. Piyush, he has joined. But
Yes, sir, he's online.
He's online, but his video is not on.
Yes, sir. Cannot pin him without the video.
Hey, Shubhda. Video on camera. Okay, we proceed further. Dr. Shilpa. Dr. Shilpa Deshpande.
Can't hear online.
I
cannot hear online. If he is in the attendees.
Okay. I invite Dr. Vandana Kate, who is the ex-vice president of Maharashtra State. She comes from Nagpur. And I request Dr. B.K. Mahavarkar to be
Hello.
the chairperson.
Sir,
to be the chairpersons for the next session. That is of Dr. Vedas Nimkar. I invite Dr. Vedas Nimkar.
Yeah. Piyush, could you please share the slides?
Yes, sir. Moment, sir.
Dr. Vedas Nimkar is a renowned consulting physician with Jupiter Hospital, Thane, and Dr. Nimkar Clinics. He was ex-secretary of IMA Thane chapter. He is first physician to conduct COVID education and awareness programs for general population at the beginning of the pandemic, and has been on multiple national TV programs as an expert. He is a clinical expert on COVID vaccination, has successfully conducted multiple vaccine master classes for doctors. We welcome Dr. Vedas Nimkar and request him to start the lecture.
Thank you, Dr. Desai, sir, for the kind introduction. I would like to thank you and the entire team of TPA for arranging these wonderful sessions today. Piyush, next slide. The talk for today is COVID-19 herd immunity, a truth or a myth? Ever since this pandemic started last year, few words have become commonplace in our day-to-day vocabulary. And the foremost was obviously herd immunity. So what exactly is this herd immunity? WHO defines herd immunity as the indirect protection from an infectious disease that happens when a population is immune either through vaccination or immunity developed through previous infection. For this COVID-19 disease, it was thought that herd immunity threshold is at around 60 to 70%. So when 60 to 70% of the population has already contracted the disease or has had vaccine, rest of the people are indirectly protected. That's what the postulate was. Next slide. Yeah. So where did this term herd immunity really come from? What is the historical context? The term was first described by veterinary surgeon Dr. Salmon in 1894, describing the health of the hogs or the pigs at a pig farm in US. There were research articles published in 1930s by Henrich et al., where the term was repeated again, and the concept of herd immunity really came of age in 60s and 70s with the success of mass measles vaccination programs in the Western world. And then came the biggest achievement or the biggest success of this herd immunity concept. In 1977, we could eradicate smallpox with the vaccination. And now comes the 20s, and again the herd immunity is in vogue regarding COVID-19. Next slide, please. Before we proceed further, we have to understand two basic concepts. Herd immunity, these two terms are integral to the concept of herd immunity. One is basic reproduction number, or the R0. It's basically the expected number of cases directly generated by a single case in a population where all individuals are susceptible. In simple terms, it's the number of other patients that one patient might infect. And the other term is herd immunity threshold, or the HIT. It is the percentage of population that needs to be immune for the herd immunity.
हाँ चलो चलो यूरिन बहुत सारे हैं ना सॉरी एमपी बहुत सारे हैं ना कैंटीन डाल
Piyush, please the background noise be shut. And next slide.
Yeah, done, sir.
So here we have a table. We've been comparing the herd immunity of various common diseases, like from measles, where the R0 is 12 to 18, which is a very high R0. One case of measles can infect up to 12 to 18 more people. And therefore, the threshold for herd immunity is naturally higher at 92 to 94%. Mind you, this was possible. The measles has to a large extent been controlled due to very successful vaccination programs. The vaccine that we have available is more than 97% efficacious against the prevention of transmission of the disease. And then the smallpox, again, the R0 is around 3.5 to 6. And it is the only disease which we have completely eradicated with successful vaccination programs. The secret there was smallpox. The vaccination induces lifelong immunity. Then influenza, the R0 is 1.3, and so is the HIT. It is lesser at 17 to 29%. I would like to make a point here. Many times we keep comparing COVID-19 with influenzas and measles. I would like to say that other than all these diseases are transmitted through airborne route or through droplets or aerosols, There is nothing else that is in common. It's like comparing chalk and cheese. Just because both are white doesn't mean they are the same. So COVID, the ancestral or the original strain, which emanated from Wuhan, the R0 was 2.9, but see the difference. The Delta variant, which is rampant presently world over, the R0 has increased from just 2.9 or 3 to 5 to 8. And so has the threshold for the herd immunity from 65% to almost 88%. Next slide, please. Last month, this was the headline in most of the newspapers. Over 85% of Mumbai population has COVID-19 antibodies, as per the Sero survey, which was conducted in the city of Mumbai in the month of August. So naturally, a lot of people started thinking that the COVID is over. And there were discussions that we have already achieved the herd immunity at a rate of 87.02% of the population in slum already being protected and 86.22% in non-slum areas. But then again, last 1 week or so, if we see, there is a small surge in the number of patients in Mumbai city. Daily, we are clocking more than 500 cases. Next slide, please. So what exactly is happening? Next slide. So why is this herd immunity so elusive? Why is it unlikely? The exact herd immunity threshold for COVID-19 disease is unknown. The present vaccines that we have, they do not prevent transmission. There are vaccine rollouts which are absolutely uneven. If we see worldwide, Western countries have covered more than 85% of their eligible population with vaccines, whereas there are so many countries in the Africa where vaccination is yet to start. If we talk about our own country, we have a state of Himachal Pradesh, which is well on the route to have 100% vaccination of eligible adults by the end of this year, whereas we have states like Jharkhand, where the vaccination coverage is less than 40%. The lower vaccine efficacy against the newer variants is well documented. There is a continuous evolutionary pressure on the virus. With pandemic raging world over, naturally there is emergence of strains with increased transmission efficiency and further immune escape phenomena. And then the pediatric population, though COVID does not really make children critically ill, but potentially the pediatric population might end up as the reservoirs of this infection. And then the immunity is not long lasting. That's what we are seeing. We'll see it in the next slides. Next slide, please. So what lessons have we really learned from Brazil? If we see the data from Manaus, which is the capital of Amazonas state in Brazil, one of the worst affected places in the world. Last year, at the beginning of pandemic, there were heart-wrenching scenes with hospitals overwhelmed, too many deaths, one of the highest death rates in the world, and no places to even bury the dead, and bodies piling up outside the cemeteries. It was really, really painful to see that. Some of you might have seen that in the news. The studies there suggested that 66% of the population was already infected in the June of last year itself, and number went up to 76% by October 2020, and the cases started to decrease. So naturally, it was hypothesized that this is due to the herd immunity. But then by January of this year, there was a huge resurgence in the cases. And then when the genetic sequencing was done, a new variant, which is now known as gamma variant or the P1 lineage variant, was documented there. In January, 100% of the cases in the Brazil, Manaus city were due to this new variant, gamma variant, which naturally had immune escape phenomenon. So in spite of having 76% or more of the population having antibodies documented, there was yet another wave due to a new variant. Next slide, please. Next slide. We have similar lessons from Israel. Israel is a country which has been a pioneer in vaccination. The country has one of the highest vaccination rates in the world, with more than 60% of about 12 years population already vaccinated by the beginning of August, and the number has since increased further. They have been vaccinated with 2 doses of the so-called one of the better vaccines by Pfizer-BioNTech, which is a mRNA platform vaccine. But in spite of this widespread vaccination, the cases started to increase from June of this year till September. And in the month of September, in the first week, Israel had one of the highest case burdens in the world amongst the 50 top countries where COVID pandemic is still raging. There were average of 833 cases per million in Israel. And by August 2021, the 87% of the sequences were of Delta. The figure was at around 20% in the month of May. So you can see as the Delta took over, the cases started to rise again in spite of the best protection with a fairly good vaccine. Israel has really been a pioneer. They have been the first country to approve the 3rd booster dose of Pfizer-BioNTech mRNA vaccine since August, and they have made it available to anyone who's more than 12 years of age. And already 1.2 million people in Israel have received the 3rd booster dose as well. And the country is well in the planning stage for launching a 4th booster, maybe 6 months to 8 months down the line. Next slide, please. So what are the limitations of present vaccines? We all know these first generation vaccines, they are not so efficient. The vaccine efficacy for the ancestral strain with mRNA vaccines, which is a new technology, but which proved to be a blessing in this pandemic. It was very high, about 94 or 95% efficacy, more than 90% efficacy against the transmission in the beginning. The other vaccines which are based on other platforms like whole virion killed vaccines or the vector vaccines, which are mainstay in our country, the figures range right from the 50% to up to 90%, depending upon the duration between 2 doses and various other factors like age and comorbidities. We have seen that the vaccine efficacy decreases with the emergence of new variants or the variants of concerns. The protection against severe disease and death is there, but not against the transmission and mild disease. The lower efficacy may lead to evolution of further mutations and emergence of newer variants. In a pandemic state, when there are sheer numbers are in the millions world over and the virus is propagating uncontrolled, there is going to be emergence of new variants and further mutations accumulating. So we must curb the overall number and just not look at just death and severe disease. Next slide, please. Piyush, next slide. Yeah, so Keener et al. From University of California, San Diego Health have showed that there is a rapid increase in the number of breakthrough infections in fully vaccinated healthcare workers in the hospital setups due to the Delta variant in US. There are naturally many issues with present vaccines, not just the scientific issues, but also the issues about demand and supply mismatch. It's really a gargantuan task to vaccinate with 2 or minimum 2 doses. Some vaccines have 1, some have 3. To the 7 billion plus population of the entire world. I mean, it's something unbelievable and unprecedented. There are a lot of cost issues there, especially for developing countries like ours. And then come the stringent storage temperature requirements for especially mRNA vaccines requiring storage at -70 degrees Celsius, up to -70, between -20 and -70 degrees Celsius, the kind of infrastructure not available in developing countries. There are adverse effects of the vaccines. They are rare. Now we have the latest figure for yesterday was 94 crore people have received at least 1 shot in India, out of which almost 25 crore have received 2 shots. So we have a large data now. The adverse effects are rare, but still they are there and they are documented. We have a news report coming in from Scandinavian countries. They have withhold the mRNA vaccines for younger population, especially teenagers, due to the heightened concerns about the myocarditis or the inflammation of the heart musculature. Next slide, please. This is something which is just in. It is a study from Qatar published by Shemeteli et al. Just about a couple of days ago in NEJM. and the finding there are really shocking. The study design is such, they have evaluated people, the population which has been vaccinated with 2 doses of Pfizer-BioNTech mRNA vaccine, and they have studied the effectiveness of this vaccine both against infection as well as against hospitalization and death 1 month after the second dose and up to 7 months. If we see the figures there, and mind you, the sample size here is very huge. Right up to in the first month, the sample size is almost 1,15,000 patients, and coming down to 1,09,000 patients having a follow-up of 7 months. So a very large and reliable data. The effectiveness of so-called one of the superior vaccines, Pfizer-BioNTech, at 7 months plus for infection is just 22.3%, and the effectiveness of this vaccine against hospitalization and death is just 55.6%. So that's something really shocking for some of us. Next slide, please. So what about the immunity after natural infection? There has been a lot of debate that the immunity that comes due to COVID infection is superior to the vaccines. And there are some other school of thoughts, people who claim that it's the vice versa. The vaccines induce better immunity than a mild disease of natural origin. If we have to talk about the immunity after natural infection, the exact duration is not known. It is believed that the immunity lasts for up to 6 to 7 months. We had discussion in Dr. Jyoti Madam's lecture about the role of antibodies and the levels of antibodies seen in people. There she has elaborated it. There are many factors which decide how long this immunity will last and how good it would be. It's up to the patient's age, the presence or absence of comorbidities, the genetics, severity of the disease, reinfection with variants of concerns, etc. The immune responses definitely do wane after 6 to 8 months. The SARS and other coronaviruses, which belong to the same family, they have shown in the previous research that immune responses to the SARS and other coronaviruses typically last at the most for 2 to 3 years. So definitely this is not a disease where we have a long-term or a lifelong immunity. And that's where the difference lies. For measles, usually once you get measles, you don't get it again. Smallpox is already eradicated, but if you really ask the old timers, once you have got smallpox in your life, they never got it again. Or with the advent of vaccination, smallpox was a non-existing entity. That is not going to be the case with COVID-19 disease. Next slide, please. So there is so much of doom and gloom since past it's been almost 1.5 to 2 years now, and we have heard a lot of bad news. It has taken a lot of physical, financial, and emotional toll on all of us. And now we have some data which says that the mRNA vaccines, which were considered superior because the protection of mRNA vaccines, which was the first vaccine to get authorized, was proved to be slightly better than the vector-based vaccines or the killed vaccines. That is also showing declining protection. So is it all doom and gloom? The answer is no. If you see that picture there should not be any fear mongering. If you see that picture over there, there is a beautiful clear sky with clouds which have silver linings. Next slide, please. So how will this pandemic really end? It's anybody's guess, but what are the ideal conditions to end this pandemic? A highly effective and cheap antiviral is found, which is effective against all the variants. There are next generation vaccines which have 95% plus efficacy, not just against the severe disease or death, but against the prevention of transmission, or so-called sterilizing immunity should be there. The vaccines need to be tweaked regularly to induce longer duration of immunity. There should be equitable distribution of the vaccines. There should not be this really Western and third world divide. There should be 100% vaccination of the entire global population, and no new variants of concern with immune escape phenomenon should emerge. But this is the ideal condition, and the world is not an ideal place. So this is not exactly going to be like this. Next slide, please. So what are the real practical scenarios? The likely scenarios, the most likely outcome of this pandemic is slowly it will cease to be a pandemic and it will become endemic. When we say the word endemic, it means there is a constant, controlled, small number of cases which continue to come, and we learn to live with those kind of cases, and it's a new normal for us with a decreasing pathogenicity of the disease. So what exactly would contribute to this scenario? The better designed vaccines, newer therapeutic options, regular booster doses, acceptance of new normal, and tweaking of the vaccines in future if new VOCs emerge. Well, this is the most likely and most plausible scenario. But there is an alternative of emergence of more virulent strains and further mutations accumulating, which make this disease more severe. But this seems to be a very unlikely scenario. Next slide, please.
Okay. Now, whereas how long you are going to continue?
Just about 4 or 5 more slides. Next slide, please.
Because time is up.
There is a case for heterologous schedules. We have asked this question time and again, can we mix and match the vaccines? The answer is yes, we should. There is data from COMCOV-1 and COMCOV-2 study is also nearing the completion. There is data that combining first dose of the AstraZeneca or Covishield, as we know in India, followed by the second dose of Pfizer BNT vaccine gives superior protection than just having AstraZeneca 2 doses. So we can see the levels of anti-spike protein IgG antibodies after this combined dosage is 12,906, which is comparable to 2 doses of Pfizer BioNTech. And then comes the surprise, the T cell responses or the memory cells are far superior than having just 2 mRNA vaccines. It's superior with Covishield followed by mRNA vaccine at 185. A similar finding has been found also in a CombiVac-S study from Spain. Next slide, please. There is a case for booster doses. Yinon et al. From Israel have published data in NEJM which says that third booster dose of a mRNA vaccine Pfizer they have used in their study decreases the relative risk of confirmed infection by 11.4 times or 11.4 fold, and there is more than 10-fold decrease in the relative risk of severe disease. Xu et al. Have published a data. It is in a medRxiv preprint. Half-strength booster dose, 50 micrograms of the Moderna mRNA vaccine after 6 to 8 months of primary series of first 2 doses demonstrated 1.7-fold and 2.1-fold rise in the levels of neutralizing antibodies against the original ancestral and Delta variants respectively. The first 2 doses of the primary series is done with 100 micrograms for this particular vaccine. So half-strength vaccine is also sufficient to give a good boost, and perhaps that can increase the number of people who can get the vaccine with the limited quantity that's available. Next slide, please.
Okay, conclude.
The primary outcomes of confirmed infection and severe illness, this is the data from Israel. As we can see over there, the severe illness, there is a comparison between non-booster group. They are the patients who have received standard 2 doses of Pfizer vaccine versus people who have received 3 doses. The number of cases over there was 294 in non-booster group and just 29 with a booster shot. And I would also like to point the adjusted rate ratio. Almost 19.5% of the patients have reported severe illness. These are the breakthrough cases, those who have been fully vaccinated. But in spite of that, almost 19.5% of the breakthrough cases did experience severe illness. Next slide, please. UAE was the first country to start with the 3rd dose. Israel, an ideal case study. It now offers 3rd dose for all about 12 years. Russia, UK, Germany, France, Indonesia, Cambodia, China, they have started for high-risk population a 3rd booster dose. Singapore and Switzerland are planning for early next year. Next slide, please. the US CDC has come up with this algorithm who really needs an additional COVID-19 vaccine. Naturally, those patients who are receiving treatment for cancers, the solid tumors, as well as the blood cancers, those who are organ transplant recipients, those who have received stem cell transplant, moderate or severe primary immunodeficiency like Sjogren syndrome, Wiskott-Aldrich, or even advanced or untreated HIV infection, or patients who are on active treatment with high-dose corticosteroids or other immunosuppressants. They have recently added the elderly population also in this group, and also the healthcare workers who, all of us, the frontline doctors who remain at the highest risk because we get still exposed daily in our hospitals and setups. Next slide, please. So what are some considerations? Why booster doses? The booster dosing may lead to the sustained higher levels of circulating antibodies. It may lead to the sustained memory cell responses. Mind you, with a booster dose, there is incremental increase in the levels of circulating antibodies as well as the memory cell responses. There are some studies, there is no meta-analysis available yet because the data is still emerging. But there are some studies which show that when a booster dose is used, the antibodies that are generated, they are broader spectrum or better antibody coverage, and they have better neutralizing capacity against the newer variants of concern. With having said all that, there is an urgent unmet need to establish the so-called coefficient of protection. We really don't know what level exactly is protective. We always say the higher the better, and that goes without saying. But exactly what is the level of antibodies, IgG antibodies against the receptor binding domain of SARS-CoV-2, that is a cutoff point. Above this, we are protected, and below this, we are not protected. We don't know that number yet. And also there is a need to correlate neutralization and protection, because as more and more people get vaccinated, it is going to be really difficult to have people who are unvaccinated. It is also unethical to conduct studies like that. So in future, to compare the newer vaccines or their efficacy, we need to have some easy clinical parameter, and which is neutralization assays, which will give us an indirect evidence of the protection that we have. So that needs to be done because we are in an unprecedented pandemic situation. We cannot really afford to have the vaccines in development for 10 years as used to be the case before this. Next slide, please.
Dr. Shashank Rishishri joined.
Yeah, I'll finish in my time. Next generation vaccines, there are saRNA vaccines or the self-amplifying RNA vaccines. Genova SGT, which is the first indigenous made in India vaccine which is coming up. It requires less dose and is stable at relatively better temperatures of 2 to 8, so refrigeration temperatures. The VOC specific boosters by Pfizer and Moderna are in development.
Can you be a little faster?
Nasally administered vaccines, they may provide better mucosal responses, better IgA responses, and thereby may provide sterilizing immunity. Two candidates by Bharat Biotech and Codagenics, which is using live attenuated virus, are in research. Oral vaccines and protein subunit vaccine. I would like to stress this one. It is
you are going beyond your limits of time. So please continue.
I will finish in my half hour. I have started late, so I'll finish in my half hour. The protein subunit vaccine by Novavax and Serum Institute of India is going to be launched in India very soon. The preliminary data suggests that it provides 100% protection from the severe disease. And this vaccine can really be a game changer. It has a lower storage temperature, 2 to 8 degrees. And this might be one candidate which might be available practically to us rather than those Western mRNA vaccines. And it might be one of the mainstays for boosters in future. Next slide, please. Advances in therapeutic options, I'm not going to discuss that. It's going to be discussed by my friend Dr. Salil later. There are molecules like PF07321332 and 8527 along with molnupiravir. These are all antivirals. Next slide, please. So the final verdict, what exactly is going to happen? Is herd immunity a truth or myth? SARS-CoV-2 is here to stay. The herd immunity with current vaccines or post-natural infection is very unlikely. The cyclical nature of disease will likely continue, leading to the further waves, but the waves might be smaller, or rather they would not be waves, but they may be just ripples. The disease severity and burden on healthcare system will reduce with successful vaccination program. Eventually, this disease may end up just like influenza or other coronaviruses after a few years. Next slide, please. The booster doses, it's not if, but rather the whole discussion should now be about when and how, because considering we have 94 crore eligible adults in India who need to be vaccinated with the first series, and almost 1/3 of them not yet got it. It's going to be really a difficult task to arrange for boosters. So discussion should revolve around when and how, but not if. Likely they are going to be annual affair, if not biannually. A lot depends upon future emergence of BOCs, their immune escape potential, transmissibility, and virulence. Off the record dosing should never ever be done. There are many friends, doctors, other healthcare workers who keep on asking, like Madam discussed in her lecture, they themselves do their antibody levels, which is not advised at all. And then they keep asking, now my antibodies have come down, should I just take another manufacturer's vaccine? The answer is no. It is worth to wait for a few months till further data is available and the clarity emerges. Next slide. This is the end of the lecture. Next slide, please. Next slide, Piyush.
Anyway, Vedas, congratulations for excellent presentation. It was very good to see you. But I want to ask only one question.
Yeah.
When you are given 2 doses of Covishield, what is the booster dose for it?
The ideal answer to that is when Covishield is an adenovirus vector-based vaccine. The protection against the transmission as well as severe disease for that is slightly inferior to the mRNA vaccine. So ideally, an mRNA vaccine as a booster dose would be the ideal case. And we have one vaccine candidate which might be launched in future. If not, the second possibility would be a protein subunit vaccine, and data for the same is expected by the end of next month from COMCOV2 studies.
No. What I was asking, when you are given the Covishield 2 doses, the third dose or booster dose now is recommended by the task force, Dr. Shashank Joshi, is Covaxin instead of Covishield. What he recommends is,
like I said, as of now, the government has not allowed the booster doses because of the ethical consideration as well as limited supplies. And there is really no role to take a third dose of same Covishield or a Covaxin. Like I said, the ideal situation is third booster dose.
May I request, because that is the topic of our next celebratory speaker, Dr. Shashank Joshi. So we rest the things with him. And thank you, Dr. Vedas and Dr. Mahavarkar, sir.
Thank you very much.
Dr. Mahavarkar is ex-president of Physicians Association and Thane IMA. And thank you, Dr. Vandana Kate, for attending and chairing the session. She is from Nagpur. And now I request Dr. Meenal Godse to take the proceedings further.
Thank you, Dr. Desai. Thank you, and Vedas.
Good afternoon, everyone. At the outset, I thank Dr. Dinkar Desai and TPA team. While we Indians are all waiting for booster dose of our vaccine, stay tuned for our next session, booster dose of COVID-19. And we have none other than Dr. Shashank Joshi as our esteemed speaker. Well, he needs no introduction. He is a prominent endocrinologist of India working at Leelavati and Bhatia Hospital and Dr. Joshi Clinic, Mumbai. He is a part of COVID-19 task force for the state of Maharashtra. He is the president of Indian Academy of Diabetes and Association of Physicians of India. He was also a former president of Research Society for Study of Diabetes in India. He is a recipient of the International Clinician Award received at 21st American Association of Clinical Endocrinologists Annual Scientific and Clinical Congress, Philadelphia. Well, many more awards are in his kitty. Last but not the least, he was honored by the government of India in 2014 by bestowing on him Padma Shri, the 4th highest civilian award for his services to the field of medicine. I welcome Dr. Shashank Joshi, sir. We are really honored to have you, sir. I also, to chair this session, I call upon Dr. Lalit Kapoor and Dr. S.V. Kulkarni, sir. Dr. Lalit Kapoor is a senior general and gastrointestinal surgeon. He is a founder member of AMC, Association of Medical Association Consultants Mumbai, a largest of its kind, which was established in year 1972, and it has crossed membership of 10,000. He is a medical director at Shakuntala Memorial Hospital. He is one of the few most sought after medico-legal consultants. He is author of book Better Safe Than Sorry, Medico-Legal Do's and Don'ts. And I'm not to forget, I'm sure you all must be reading his article in GRASP. I'm getting a feel-home feeling while introducing Dr. S.V. Kulkarni from Amchi, Navi, Mumbai. He's a senior consultant.
Yeah, I'm there. I can see you. Thank you.
Medicine and Digital Health in India since 2011. He's the founder member of API Concord Chapter, Raigad Medical Association, and Maharashtra Association of Physicians. Well, there was really much more that I could have added for all these three dignitaries, but the time doesn't permit me. So I welcome you, all three of you, and let's start for our talk. Yeah, thank you very much once again.
Thank you, Meenal, for the wonderful introduction. Nice to see Shashank after a long time. After the fantastic introduction by Meenal, I will describe Shashank as only one word, that is intelligence personified. And he has been certified by the Prime Minister of India also for his Vicharonki Shuddhata. He is one of the most sought over speaker, and he is going to have answers of all the questions which we are going to have in our mind. His and Dr. Harish Shetty's work has been absolutely exemplary for the COVID pandemic. So please welcome the past Dean of Indian College of Physicians and the Southeast Asia Chair for International Diabetic Federation, the darling of crowds of all academic meetings, Dr. Shashank Joshi. Shashank, please.
Thank you, Dr. Kulkarni and Dr. Godse, for your kind words of introduction. I must pay my regards to Lalit Kapoor, sir, who has always been guiding the medical profession for the last 4 or 5 decades. And I must thank Dr. Desai. I must first congratulate Dr. Vedans for an outstanding presentation. I think he was very crisp, and I think he got interrupted, unfortunately. And, the topic of mine given to me is booster. Let me take off from where he left. He said that boosters are going to be needed, but the only issue is when and how. So let us look at this booster issue. First thing is that all of us have received at least 2 doses. There is only one exception to that, which is the Johnson & Johnson vaccine, which is a single dose. That is still not available in India. And there are exceptions like the Zydus Cadila vaccine, which is not easily available. There are 3 doses. After that, a booster dose is expected after 6 months. And that booster dose in the US, FDA has approved it for all people above 65, for healthcare professionals, for frontline workers, as well as between 18 and 65 to people who have moderate or severe immunocompromised conditions. It could be a cancer chemotherapy patient, it could be a patient of dialysis, it could be a patient of solid or hematological issues. It could be immunological diseases of serious consequence. It could be HIV/AIDS with severe immunodeficient state, so on and so forth. So this has become a standard of care as far as United States is concerned. But remember, every vaccine which is there on planet Earth is emergency use authorization. And all these are experimental vaccines. The degree of the protection they confer is not very clear. Currently, the early generation vaccines confer only 2 things. Number one, they prevent deaths and they prevent severe infections, but they don't prevent COVID. So that is the first thing which we need to recognize. This is the first thing we need to recognize. The first thing which we need to recognize is that we need to recognize that our currently available vaccines are all early generation. They confer protection only for against severe death, and against severe diseases. That is the only thing which we know. Now, when should we advocate a booster? India has a population of 139 crores. Currently, we have vaccinated 94 crore Indians. We are waiting for a milestone this week. And the government of India needs to be congratulated that this is the largest vaccine program on planet Earth. And 100 crore Indians will be vaccinated at least with a single dose. We are committed to vaccine equity, which means vaccine samata. And therefore, till the second dose is administered to a large population of India, or 100 crore Indians at least are vaccinated, it is unlikely that a booster recommendation will be made by NTAG. And Dr. Arora sir, who is the chairman of the NTAG, and the chief of the National Task Force, Dr. Vinod Paul sir, have clearly given a verdict that they are waiting for a large Indian population to be vaccinated first. Our commitment is to save every life due to COVID. We are I pay my homage to the 1800 doctors who have lost their lives. So many healthcare workers have lost their lives due to COVID. But it is very likely that after the 100 crore milestone will be done, booster will be recommended. Now, the biggest question is which booster, how frequently, and to whom. It is very likely that the booster is going to be given to frontline workers, healthcare workers, moderate to severe immunocompromised state in the age group between 18 and 60, and above 60 probably for everybody. That hierarchical preference will be based on availability of vaccines currently available in India and based on the vaccination status. This likely guideline is coming probably post Diwali or after that. So we don't know when it will come, how it will come. That is unanswered question. Let us look at the science of booster. The science of booster is very clear cut. The science of the booster is very, very clear cut. The science of booster is that we definitely do not get adequate protection after 6 months after 2 doses. And therefore, it is very, very clear that we need to get a boosting dose. Now, in India right now, most of us are being given Covishield, and the small group has got Covaxin. Now, a question has been asked, should we do mix and match? ICMR had only one anecdotal report from Gorakhpur, where accidentally Covaxin was followed by Covishield, which gave a better antibody response. Spain, United Kingdom, all have shown that when we mix and match vaccines, clearly we can understand that whenever mixing and matching is done, the response is better. That data is very clear cut. Now remember, only antibody status, Dr. Vedant very nicely said, is not sufficient to know how protected we are. And therefore, in that sequence, we need to understand two things. That we have to look at T cell immunity and B cell immunity. So remember that the Covishield or the adenovector-based vaccine, which is Covishield, the Sputnik, as well as other vaccines, they all are fundamentally causing the T cell immunity response very good. Antibody response is very good. Covaxin is generating reasonable antibodies, may not be as robust as the Covishield antibodies. But if you mix and match, probably you get a very good response. The mRNA vaccines, which are not available in India still, and as Dr. Vedant said that we might get availability from Genova, one Indian vaccine, the Pfizer and the Moderna, even after 2 doses, all of them, their antibodies fall away. Usually we have seen with 1 dose of vaccine, the protection will last for 3 to 6 months as far as the spike protein antibody is concerned. My humble folded hand request to each one of you all in the audiences, please do not do antibody testing. In USA, Europe, antibody testing is not available. It is only available for research setups. And if at all you do the antibody levels, then either do it against the RBD or the nucleocapsid or the virus neutralization test, which very few labs do. Because the PNRT method by which we test vaccine efficacy is not known. We will need boosters. There are no two ways about it. Remember, our children population also needs to be vaccinated. So we have to ensure that a large Indian population has to be saved before the booster has to be administered. But we also need to save the lives of the people once their antibody response declines. So unless and until you are into a research trial program, currently there is no recommendation of doing antibody status. But if you are immunocompromised, if you are having moderate to severe immunocompromised status, currently there are better treatment options available. We will soon have the launch of Molnupiravir, which is a better version of Favipiravir. It's the first anti-COVID drug on planet Earth. All other drugs were repurposed drugs. Remember, favipiravir was a flu drug for Japan, and this is an anti-COVID drug which will be available. We have the advent of monoclonal antibodies now, the Lytic cocktail, which is also very robust and strong. So obviously, now there are better treatment options available. I think it is just a matter of a month, after which officially the booster dose will be recommended. Please do not rush for a booster dose. If you have a specialized disease, which allows you to have moderate immunocompromised state, only my request to everybody here is that be little patient, wait for the official guidance. And if you are in a situation, even today, for all variants of concern, the best vaccine is double masking. Ensure that you either wear an N95 mask, or if you're wearing a cloth mask, use a 3-ply surgical mask, which is 2 rupees with a cloth mask. That will take adequate protection. COVID is an airborne virus. We have seen a lot of breakthrough infections. Most breakthrough infections have been mild. Remember, therefore, don't rush. Data on vaccines is very, very premature. We are a long way away from the data on vaccine generation. So do not rush to run for a booster, because official guidance will come once they study the Indian data. Once the Indian data is sufficient, Indian DCGI, the Indian NTAG, the Indian task force feels it is appropriate to recommend a booster dose, you will get the guidance very soon. Also, they have to balance vaccine equity as well. So it's just around the corner. Till that time, mask yourselves appropriately, ensure that you stay protected, and be careful when you are in a crowded environment. That is all I have to say here. Remember that we'll definitely need boosters in the times to come. Probably it will be every 6 months or 1 year, we don't know. The data is emerging. This is the fastest developed vaccine in the viral arena or the microbial arena. No vaccine has been developed within a span of a year and a half. We are only 2.5 years in the pandemic. So remember that when the early generation vaccine has at least saved lives, at least saved severe disease in most of the time in most of the people. Individual exceptions will always be there. So please don't tell me that 2 vaccines were taken and someone died. You could always get immune escape. Immune escape is known, but it occurs only in 0.1% of this. So rarely you can get these problems. So my request is you will get the guidance. Healthcare workers were given a priority by our Honorable Prime Minister in the vaccine program. We were the first group to be vaccinated, followed by the frontline workers, followed by the senior citizens and the comorbidities. Very soon you will have a guidance coming for a booster dose as well. Which booster, what mix and match. We are also developing a nasal vaccine. The nasal vaccine is also undergoing trials. So that will also come. Pediatric vaccines are being developed. So be patient. And as I said, currently our best booster is double masking, and the new booster guidance will be available in a month from now. Thank you for the opportunity given to me, Dr. Desai. And I'm certain that there will be a lot of questions on this very, very interesting topic, because there are a lot of unknowns. COVID has only one thing which is clear-cut. It is predictably unpredictable. And data related to COVID is also predictably unpredictable.
Thank you, Dr. Shashank Joshi, sir. Dr. Lalit Kapoor, sir.
It's indeed a great pleasure and a privilege to be at such a wonderful conference organized by Dr. Dinkar Desai, who I know is a great innovator, a great organizer, and in fact, you don't see a lot of pathology conferences, and is extremely well organized. And on top of it, we have I'm chairing a session by such a great person who has done tremendous amount of service to the not only to the medical profession but to the society at large. And I can say that despite being so busy and so much occupied with a lot of administrative and other things, he has been extremely kind, and whenever we personally phone him for a personal query, he has really gone out of the way and answered the queries which are so reliable, and therefore we like to hear from the horse's mouth. Of course, this topic was very relevant, particularly to me. Now, I had a moral dilemma which I would like to share with Dr. Shashank Joshi, and maybe, of course, he's, I think, already given the answer. Some influential doctor friend phoned me and said that, look here, do you want the third booster dose? I can arrange for it. many people are taking. So I told my wife, and she said, why not? If we are getting the third dose, why not take it? But then when I just spoke about it at my house, my daughter overheard me. And she said, how mean you are. How can you take the third dose when people have not even got first dose? People have not got second dose. And how can you take? And so immediately I decided that nothing doing, even if somebody is giving me the third dose, I shall not take it. So this was a dilemma, which I think Dr. Shashank Joshi has quite clearly said that please wait. And it's something that going to happen soon enough, but it's not in the interest of equity. It is not good to deprive people who have even not got the first dose. So I think this is a great message which Shashank has given to all of us, because I know there are several people who have received the third dose or the booster dose and have found ways of getting it done. But I think one needs to have certain considerations for society at large. So it was great being here and great hearing Shashank, who has dealt so beautifully with the media. I am very impressed with the way he has dealt with the media. He's called a spade, and I heard him told one reporter at the NDTV quite bluntly that she was unnecessarily spreading panic by saying that children might be affected, they should be given vaccine and so on. So I think he's a person who has truly and very well represented the profession and done great service. We are honored to have a person like him looking after the COVID issues. Thank you very much.
Dr. S.V. Kulkarni, sir.
Yes, thank you, Shashank. You have clarified and demystified all the doubts of everybody, and I really appreciate your enthusiasm, your digital presence. You can be only compared to Lata Mangeshkar or Sachin Tendulkar only. You are so famous because of your knowledge and the confidence with which you deal with all the situations, and you make everybody so comfortable. Under your leadership, we are sure that we are going to win this war. And thanks for clearing so many points in the minds of all our physicians and all the pathologists who are attending. It's a wonderful lecture as usual. Thank you.
So thank you, Dr. S.V. Kulkarni, President of Navi Mumbai Physicians and Medical Association, Dr. Lalit Kapoor, sir, a very renowned personality in medical field, and Dr. Shashank Joshi, for giving a consecutive second lecture in the Pathologist Association's forum. And thank you, Meenal, for going with all this master of ceremony job. I request Dr. Supriya Lad Chinsolkar to take the further proceedings. Dr. Supriya Lad.
Yes. Good afternoon. Thank you, Dr. Desai, sir, for giving me this opportunity to introduce wonderful speakers for the next session. The next session is a very important topic which continues our COVID theme. It is about COVID management, lessons learned from the second wave. And it gives me great pleasure to introduce our speaker for today, Dr. Anand Bhave. Dr. Anand Bhave, sir, welcome. He is a consultant physician at Param Hospital and Jupiter Hospital, Thane. He is the member of the Thane COVID Task Force. He is the ex-honorary professor of medicine, University of Mumbai, ex-chairman, scientific committee, MAPCON and RSSDI, Maharashtra State. He has presented scientific papers in various conferences, delivered lectures in conferences and IMA meetings. He is the life member of the Association of Physicians of India, the Research Society for the Study of Diabetes in India, and the Indian Medical Association. And to us Thaneites, he is Dr. Anand, who is always there for us, always a phone call away. And to conduct this session, I would like to invite eminent chairpersons to conduct this panel discussion. Dr. Anand Kate. Another Dr. Anand, gives me great Anand to introduce both the Anands. Welcome, sir. Dr. Anand Kate from Nagpur. He's done his MBBS and MD from Government Medical College, Nagpur. He's a practicing physician at the Ameya Hospital, Dharampet, Nagpur, since 1989. He's a senior consultant in critical care and internal medicine, Wokhard Hospital, Nagpur. Obviously, we have a very esteemed panelist here. Sir is a past president of the IMA Nagpur branch, past president of the API Vidarbha chapter, CWC member, IMA headquarters, chairman IMA-MS for the professional protection scheme. He has delivered the CL Zaveri oration on the chikungunya at the IMA state conference, which was held very much in Thane in 2007. And of course, he has delivered many more lectures in different forums. Many awards to his credit. He's been the best secretary in 2003, best president of his branch in 2006, and best branch award at the national level in 2006. And joining him, I would like to invite Dr. Shilpa Gupta, who is a physician and intensivist at Hiranandani Hospital and the Thane Healthcare Hospital. What an eminent group here. It gives me great pleasure, real Anand, to invite Dr. Anand Bhave and Dr. Anand Kate. So I would request sir to
Dr.
please
Supriya,
today's
you
topic.
can introduce Dr. Shilpa also, Dr. Shilpa Gupta.
I did that, sir. I already did. Should I say again? Dr. Shilpa Gupta, who is physician and intensivist at the Hiranandani Hospital and the Thane Healthcare Hospital.
No, thank you. Okay. I missed it.
So interesting topic for now, COVID-19 management, lessons learned from the second wave. Over to you, Anand, sir.
Thank you, Dr. Supriya. I'd like to share my screen now. Basically, it's important to remember that unless we learn lessons from history, we are bound to repeat the mistakes. And that is why after any major event such as the second wave, we need to take home some lessons and apply them so that we can manage such disasters better in future. So lessons from the second wave are many, and I would just cover a few of them. There are different types of lessons. There are clinical lessons, there are management lessons, even philosophical lessons. The first one, I would say, is to beware of what is called the exponential curve. The exponential curve looks like this. And this is how a wave builds up. So every COVID wave starts very small with small number of cases, 5, 10, 15, 20 cases, and they don't trigger an alarm. And for almost 6 or 8 weeks, the number of cases remains small until suddenly the system is overwhelmed when a tsunami of cases hits society. This is because there is an incubation period when an infected person infects somebody else. The other person does not manifest symptoms and signs immediately. And then after a 4 or 5 day incubation period, then the symptoms are noticed. By the time that other person has started infecting some of his contacts and so on. So the disease course is also a little slow. For the first 6 to 7 days, generally patients do not become seriously ill. And those who develop pneumonia or cytokine storm, they become ill in the second week of the illness. So by the time the hospitals start filling up and ICUs start getting overwhelmed, the wave seems to appear as if it's a surprise. But one must always remember that waves start small. And that is the reason why some countries like Australia, New Zealand, we read in the papers that even after 10 or 15 cases, they hurry to force a lockdown. So we must always keep watch on the number of cases and not wait till the cases start shooting up. The second important lesson, according to me, is to keep politics and bias away from science. Unfortunately, even though COVID is totally neutral, It kills everybody, whether you are right wing, left wing, or you're from this country or that country. Human beings have a habit of inserting politics into everything, and unnecessarily that interferes with science. It interferes with data collection. It causes people to deny things. There are competitions to claim credit on who has managed COVID better. There are competitions to hide the number of cases, and there are comparisons between states and this government, that government. That is all nonsense. COVID is a threat to all humanity, and we have to come together and fight it. For example, biased reporting or politics can interfere with data collection. Now, how big was the second wave actually? Now, although the official data collection reports around almost 4 lakh deaths in India and a certain number of cases, it has to be accepted that India has huge problems. It's an overcrowded country. It's the second largest populous country in the world. And everybody expected that COVID will hit India. And it is not to a discredit or credit of any government or anything. The things have gone as predicted. Every country in the world has underreported number of cases, whether because patients are unwilling to test, or whether they are not tested, or the doctors are late to test, for whatever reason. But even though our number of deaths due to COVID was reported at around 4 lakhs, there are several experts who do feel that the big wave was much bigger than this. The Center for Global Development in Washington conducted a study which found that the total number of deaths from all causes, not just COVID, all cause deaths, since the beginning of the pandemic is almost 40 to 50 lakh excess deaths in this period compared to the pre-pandemic period. And this was a major organization headed, one of the panelists was Mr. Arvind Subramaniam, who was former Chief Economic Advisor of India. Similarly, this other data conducted by our own ICMR, they conducted surveys. How many people in India have antibodies to COVID? In May and June 2020, just 0.7% of Indians had antibodies to COVID. And we had a very strict and effective lockdown for most of 2020. So the seropositivity rate, again, it was tested in August, September, was just 7.1%. But by the end of the year and beginning of this year, 2021, it had risen to 24%. After the massive second wave hit India, a huge percentage of population was infected, and the latest Sero survey in June-July 2021 shows that almost 2/3 of all Indians have now got antibodies against COVID, which indicates that the number of infections was far, far higher than the number of reported infections. And there is no credit or discredit in this. But if we start, arguing for or against any particular party or government or things like that, then we tend to argue about basic data. We confuse the picture, and if we don't have a proper idea of number of cases, number of deaths, then we can't manage the pandemic better. So we have to keep politics and bias out of science. The third important lesson is to maintain basic supplies and provisions. The second wave was so large that we all remember those days where people were unable to get oxygen. They were looking around everywhere for oxygen beds, oxygen concentrators. People were not able to get remdesivir and many other drugs. Then when the mucormycosis wave started, they were finding it difficult to get antifungal drugs. We have to remember that this is a highly unusual situation where suddenly large numbers of people are falling ill, and no healthcare system in the world. We saw that there was overwhelming of the healthcare system, not just in India, but in US, UK, everywhere. So this is not something you can plan for before. Second wave, now we are much better off to deal with the third wave. Number of ICU beds, number of oxygen cylinders, the government has done a lot about maintaining oxygen supply now. Many of the hospitals which are more than 50 beds in strength, have been asked to purchase their own oxygen plants. The smaller hospitals have been asked to sign contracts with the oxygen suppliers to ensure the supply. And in fact, the municipal corporations of Mumbai, Thane have set up their own oxygen generating plants. I'm sure if the wave hits any time now, there will not be such a shortage of remdesivir at least and several other basic drugs. The number of healthcare workers is a very essential resource which cannot be generated overnight. But this is the most important lesson from the second wave, to be ready with the provisions and supplies for the third wave. And as a result, we have still maintained large COVID jumbo hospitals, even though most of them are empty right now because of the low number of cases after the second wave. For clinical lessons, the first lesson is to avoid an over suppression of immunity. What has happened, what we have seen, is that probably in many hospitals, in many centers, there might have been excess use of steroids, both in terms of dose as well as duration of steroids. The excessive use of tocilizumab and with added to the immunosuppression that COVID itself creates. We have seen a number of cases of mucormycosis, and not just mucormycosis, but invasive fungal infections like aspergillosis. There have been cases after discharge of COVID that activation of tuberculosis has happened. So all these are consequences of over suppression of immunity. We have to follow the recommended doses. For example, it is just 6 or 8 milligrams of dexamethasone per day and only 80 to 120 milligrams of methylprednisolone, whereas we do see some cases where they have received mega doses or pulse doses like 500 mg of methylprednisolone per day, which have no added benefit and only added risk. Another lesson for the clinicians is to be very careful about diabetes. There seems to be a very close relationship between COVID and diabetes in many ways. COVID seems to even precipitate new onset diabetes, and we have seen already, all my colleagues must have seen that maybe those who were pre-diabetic or prone to diabetic before have landed up as frank diabetic after they have recovered from COVID. The sugar levels do worsen rapidly after getting serious COVID, especially not only due to the COVID itself. Maybe there is a damage to pancreas, maybe there is a gross increase in insulin resistance, and of course due to heavy use of steroids, it becomes difficult to control the sugars, and insulin becomes the mainstay for admitted COVID patients. Probably those people who are on SGLT2 blockers may benefit by stopping those drugs for a few days if they have contracted COVID, because these drugs may be associated with an increased risk of ketoacidosis and what we call as euglycemic ketoacidosis, where the blood sugar levels may not be very high, but still the patient ends up with ketosis. The high incidence of mucormycosis is particularly related to cases which have diabetic ketoacidosis as a complication. Restrict the use of antibiotics. Another clinical lesson is this: every COVID patient practically ends up getting some antibiotics without any benefit of their use. Especially in the OPD, you will see 2 or 3 antibiotics, and the thing becomes even worse for patients who are admitted right from day one for absolutely no reason. They are given higher IV antibiotics, and then later on, if that patient requires steroids, requires tocilizumab, lands up with serious sepsis, there are hardly any antibiotics available to use because the higher antibiotics have been used already. If this continues and we repeat all this in the third wave, then we are going to lead to this situation that a post-COVID era will be the era of superbugs, bacterial infections which will be resistant to all antibiotics, and we really don't want that. The next lesson that we must remember is to anticoagulate properly. Still, there are many centers who are not properly anticoagulating their COVID patients. COVID and coagulation go hand in hand. The virus itself infects the endothelium and damages it. Inflammatory cytokines, the active phase reactants, the complement, they also end up damaging the endothelium. And this disturbs the workhouse triad, setting up a pro-inflammatory state in the body. So the patient may land up with pulmonary thrombosis, not just pulmonary embolism, but in situ microvascular thrombosis in the lungs, cerebral venous sinus thrombosis, microvascular arterial thrombosis with what are described as COVID toes, limb frank ischemia in the limb, myocardial infarction, large vessel occlusion, strokes with paralytic attacks, clotting of your central venous catheters, and of course the commonest one is DVT, which may affect more than 10% of all admitted ICU COVID patients. So this is a very big issue. You will find this has been discussed repeatedly now. A lot of attention has been given to this. And the August issue of Journal of Association of Physicians of India does discuss this in detail. Practically, number of patients who are admitted are getting low molecular weight heparin, and most of them are getting some short course at least of a drug like rivaroxaban or other oral anticoagulants at discharge. One of my friends from Pune, a pediatrician, Dr. Avinash Kinikar, has repeatedly raised the issue that so far we have not given any guidelines as to how long after discharge from COVID should a patient begin to exercise, should go to the gym. And the reason is that if the procoagulant state persists even after discharge, are these patients at excess risk of suffering a myocardial infarction? We definitely need to set up guidelines on that also. A very important point for all physicians and indeed all doctors is that we have begun to see number of cases of multi-inflammatory syndrome, multi-system inflammatory syndrome after COVID, even in adults. The syndrome in children has been well described, and the pediatricians are very well tuned to detect this syndrome of inflammation, which occurs in children after they have suffered from COVID, which may be totally asymptomatic. But now the second wave, which has infected so many adults, has thrown up cases of adult MIS, and even in our own setup, myself and my partner, Dr. Sita Raju, we have seen almost 4 to 5 multi-system inflammatory syndrome in adults. And recently, the latest issue of JAPI has reported a series of 4 cases of adult MIS. So we need to discuss this in detail, and all clinicians need to remember this. Whenever we get a case of fever, obviously we first think of things like malaria, dengue, leptospirosis, and now pneumonia has hit our area of Mumbai and Thane in a big way. We think of typhoid, but now at the back of our mind, a case of fever going on for a week or 10 days, all these tests turning out to be negative, we must think of adult MIS. So how would you define a case of adult MIS? These criteria have been given by the MMWR, that is morbidity and mortality report categorization. If it's a severe illness requiring hospitalization in adult older than 21, you must consider MIS. Of course, the WHO will consider even a milder illness not requiring hospitalization in this criteria. The patient must have some evidence of COVID either currently or in the recent past. So usually they say that within the last month or 2 months, or at the most 3 months, if a patient has got a history of COVID from which he recovered and now landed up with fever. Sometimes the patient may not even remember that he or she had COVID. It may have been asymptomatic. In that case, the patient should have a clear-cut positive COVID antibody. And of course, if the patient has got a Covishield or a spike vaccine, then if the patient also has antibody to the nucleocapsid N protein, that indicates that this antibody has come from infection and not from the vaccine. Then the patient should have at least one or more extrapulmonary organ system involvement, which may be in the form of shock, that is hypertension, or cardiac failure, arterial or venous thrombosis, liver dysfunction in the form of abnormal liver enzymes, or renal failure. There should be lab evidence of severe inflammation, that is very high CRP, ferritin, D-dimer, and interleukin-6. And what distinguishes COVID from severe COVID from MIS is that lung involvement is very mild or it may be absent. At the most, there may be a mild effusion. If there is a severe COVID, then the disease starts mainly from the lungs. You cannot have a severe COVID patient who has no pneumonia. Every patient of severe COVID has to have GGO or pneumonia on X-ray or CT scan with hypoxia, because the disease probably starts from the lungs, mainly attacks the lungs, and then causes systemic cytokine storm. But in MIS, the lung involvement is relatively mild or even absent, and the other systems are strongly affected. Another distinguishing factor is, of course, the RT-PCR positive goes in favor of active severe COVID. But remember that some cases of MIS may have even RT-PCR positive. The lab evidence, the CRP, ferritin, etc., are much higher in the MIS than severe COVID. And basically, when you are going to diagnose MIS, you must first exclude other serious infections like bacterial sepsis. This can be quite difficult to exclude bacterial sepsis. Why? I will just come to that. Now, features of adult MIS usually seen 3 to 5 weeks after COVID, but maybe up to 12 weeks also. And the gap between COVID and MIS can be difficult to estimate in most cases. It is probably a hyperimmune post-COVID response. And again, there have been speculation that there may be persistent virus. The COVID virus may persist outside the lung, maybe in the intestine, and that may be causing or driving a persistent immune response. If the immune response doesn't switch off, there is a persistent elevation of IgG antibodies, which stimulate the monocytes especially and the macrophages. Then also you may get an MIS. Most of the reported, or in fact all reported adult MIS cases were below the age of. So an elderly person with a PO, you would less likely suspect adult MIS. And in fact, majority are below the age of 35. The reason for this is that MIS is an hyperimmunity disorder. It's an immune storm, and it happens in basically healthy people who have a strong immunity, and not in elderly people with a suppressed immunity. The presentation, usually the commonest signs, symptoms are fever, myalgia, and just like pediatric MIS, abdominal pain can be a very prominent symptom, as it was indeed in one of my patients. Eventually, the patient may land up in hypotension and require noradrenaline drip. Patient may go into renal failure, but often this renal failure is not extremely severe. It does respond to fluid resuscitation and to support of blood pressure. Liver enzymes are elevated. There might be altered lowered serum albumin, high bilirubin. Just like pediatric MIS, some of the adult MIS may have cardiac dysfunction, which is the most dangerous manifestation. As at least whatever we saw, some of the deaths were more due to cardiac dysfunction than the other organ dysfunctions, which could recover. But sudden cardiac failure, pulmonary edema, Sudden arrhythmias can be fatal. Lung involvement, as I said, is usually mild in the form of pleural effusion, and there may be lymphadenopathy, which is more common in pediatrics, but can be there in adult MIS also. The lab features, as I said, there has to be some evidence of COVID in the past in the form of antibodies against COVID. WBC can be normal or elevated. And as I remember, in our cases, they are mostly elevated. Platelets may go down. Creatinine and BUN elevation alter liver enzymes. Now, to confuse the whole picture, procalcitonin also is often strong positive. So this leads to a suspicion of bacterial sepsis, and most of such patients may end up getting antibiotics first. But if you have repeated blood culture negative, no source of bacterial sepsis, and still PCT is positive, WBC elevated, and very high markers of inflammation, then you must suspect COVID as one of the causes of PO in the form of adult MIS and do the test. Obviously, to diagnose adult MIS, the other causes have to be ruled out, like malaria, dengue, chikungunya, and leptospirosis. CT chest and abdomen may be normal, or there may be an effusion. And cardiac markers like TROP-I, CKMB, or NT-proBNP may be elevated, and a 2D echo may show cardiac failure. Treatment of adult MIS, now all of us will need to be familiar with this because we will be seeing cases. Again, it's an immunological disease, and steroids are extremely important in this. And the dose of steroid required for MIS would be higher than what we normally use for acute COVID. Anticoagulants are required, initially low molecular weight heparins. And because you can definitely see a lot of thrombosis in an MIS, tocilizumab may be required. Interleukin-6 is elevated, and patients are not steroid. The options are tocilizumab versus immunoglobulins. Immunoglobulins have a very good record of success in the pediatric MIS, and in adult MIS, definitely they have an important role. The only problem for adults is that it's a very costly drug and definitely difficult to give for most of our patients. And obviously, these patients often land up in an ICU. They require inotropic support and support for all the organ systems. So this is one of the important lessons, that be prepared for more and more cases of adult MIS. Thankfully, it is still extremely rare, but not so rare that you can forget about it. After all these sort of depressing lessons or negative lessons, there is one good lesson also, that reinfection is quite uncommon. The second wave was a huge wave which infected so many people, but we all saw that it infected in 2020 in the first wave were not infected again in the second wave. So this does not mean that reinfection cannot happen at all. It can. In fact, right now I have a friend who has been infected again just now in spite of a past infection and 2 doses of vaccine. So it can happen, but still it is extremely uncommon or rare, because post-infection natural immunity, at least in my opinion, seems to be stronger than the post-vaccine immunity. There have been many studies about reinfection. There was a big study from England where they studied that people who were infected in the first wave, the healthcare workers, how many of them got reinfected in their second wave. And they found that the reinfection rate was just 0.86%. And importantly, they saw that even if there is no antibody titer detected, but you know that the patient has a history of prior COVID, still there is a protection against reinfection. Then there was a massive population level study in Denmark, which showed a reinfection rate of just 0.65% in the next wave. However, in the elderly group and those with comorbid conditions, the chances of reinfection could be higher. As Dr. Vedas rightly said just a couple of lectures ago, that the immunity from COVID or natural immunity from COVID may not last forever. In fact, looking at other coronaviruses, it may wane after a few months or even may go up to 2 years. We don't know. It's still study under evaluation. But definitely, this is the way that the pandemic subsides is after all this, that people don't keep getting infected again and again in every wave. And that is how the pandemic is going to subside. So natural immunity is our savior. Looking at all these lessons, apply that to the third wave. What will be the third wave like? Now, there are many sort of points about the third wave. First of all, will there be a wave? Some of you may be knowing that just last week, the Bombay Municipal Corporation told the High Court of Mumbai that there is not going to be a third wave based on their own data, that in Mumbai, the seroprevalence of antibodies is almost 87%. Most of the healthcare workers, up to almost nearing 90%, have antibodies already. And in fact, even children have a large proportion of them do have antibodies. So there may not be such a big wave because already many people are immune in Mumbai. But even if 86% are already having antibodies, Mumbai has 1.2 crore citizens, and there's still a vulnerable population of more than 20-25 lakhs. So there are still enough number of people being infected, and they can call a wave. When will a wave come? Now, this is also a sort of a speculative topic. There are mathematical models which study this issue, and there are many experts who predict when the next wave will come. But to my mind, a wave starts when a lockdown is lifted. It is as simple as that. So now, as the lockdowns are being lifted, now schools are going to open, malls will open, cinema theaters are going to open. Then the first point that the wave starts building up as an exponential wave. So initially there are going to be very few cases, and within 6 to 8 weeks, that is about 2 months after lockdowns are lifted, the wave is going to come. So that doesn't require rocket science. When you remove the lockdown and unlock the population, the wave starts. It's as simple as that. Will it affect children predominantly? There was a lot of talk in July, August that the next wave might affect children predominantly because schools will open, etc. But there are many points against this. First of all, children have mild disease. So it's not like, I mean, children will get infected, but it's not like they are going to fill the ICUs so much. Thankfully, even if there will be some serious cases, Mumbai, Thane Municipal Corporation has prepared ICU wards for children. But basically, it's not a serious disease of children. Secondly, it's not as if children are infected so far. What has happened is that they have been asymptomatically infected, and the sero survey conducted by ICMR showed that almost 60% of children in their survey have antibodies. So they have already been infected to a large extent, and so it's not that we have a very huge population of only children who are now susceptible. Will the third wave be deadlier or will not be deadlier as compared to the second waves? To decide this, we have to see that the third wave is, now this is a point about children, that already 57% of children in the 6 to 9 age group have antibodies. ICMR has recommended starting primary schools based on this data. Children, by default, they have mild disease, and this has already been seen in the second wave. There were cases where parents got infected. They panicked. They didn't want the children to get infected, said to live with the grandparents, which was a mistake. In several cases, the children were already infected. They were asymptomatic. and they went and passed the infection to the grandparents. So the teaching by pediatricians is that if the parents are infected, do not send the young children away. They can stay in the same house. Schools being restarted will definitely lead to some increase in cases, but hopefully not so many ICU admissions. Will the wave be deadlier as compared to the last one? Well, vaccination numbers before the second wave were not very high. It was only healthcare workers, frontline workers who had really been vaccinated by the time the second wave started. Now the situation is changing, and as Dr. Shashank said just a little while ago, 95 crore Indians have got at least one dose. So hopefully that will reduce the number of serious cases at least, and the wave may not be deadlier. Lots of people have already been infected, as the ICMR data shows, and they are likely to have either no disease or milder disease. We are far more aware this time. The infrastructure will be better. The awareness is there. Oxygen plants, drug supplies will be better. And another thing which is different if the third wave comes are the new drugs and new trials are becoming available. This topic will be covered by the last speaker, so I'll not elaborate, but already many of us have used monoclonal antibodies for some of who are at high risk. Inhaled budesonide has now become part of the protocol, and the AIMS ICMR protocol mentions this as one of the options, which may hopefully reduce the incidence of serious disease. The new antiviral kid on the block, molnupiravir, trials are already being held even in India. And this drug, if it is able to work as they have claimed, it may hopefully reduce some of the mortality, morbidity. And with baricitinib and tofacitinib in the ICUs has enabled our intensivists to treat the serious cases better. So hopefully there may be a wave, but it will be milder, or at least it will be treated better. And as time passes, even if there are new mutations, hopefully they will not be deadlier and deadlier than before, and eventually COVID will become an endemic disease, which we will not panic, though we have to be careful all the time and still continue to wear masks. Thank you.
Thank you, Dr. Anand Bhave, for giving an excellent talk on how we manage the second COVID wave in our country and the lessons we have learned. Definitely, when the second wave was in progress, the situation was really alarming and overwhelming, and everybody was running from pillar to post. But it's absolutely true that when we treated patients in the first wave, we hardly knew anything about the COVID and all the protocols, treatment, and it was a continuous learning process for all of us doctors. And by the beginning of the second wave, or maybe mid of the second wave, I think we had sufficient knowledge to treat the patient. And the other points which you have covered were really very much interesting. One thing I think what we have learned and what we have shown to the world by treating the second wave of COVID is that how to treat, how to tackle a pandemic in resource-limited circumstances. I think we had limited resources, as you had already mentioned. Drugs were not available, oxygen was not available, beds were not available, but still we could manage and we could control the wave in appreciably short period of time, and that is really very good feature about the medical fraternity in India, and everybody is lauding it all over the world. Thank you, Dr. Bhave, for an excellent lecture. Before I stop, I must thank Dr. Desai for giving me this opportunity to chair the session today, and of course, Dr. Supriya for giving a wonderful introduction. Thank you very much.
Dr. Shilpa? Dr. Shilpa Gupta? Please
Hello?
unmute. Hi.
I am unmuted. Thank you so much, Dr. Anand Bhave, for such a nice lecture. It was very informative. Thank you, Dr. Dinkar Desai, for giving me this opportunity to be here. Unfortunately, I'm in hospital and I joined at the last moment. Thank you, sir.
Thank you, everybody. Dr. Anand Bhave, Dr. Anand Kate, Dr. Shilpa Gupta, and Dr. Supriya Lad for making the session very live. I request Dr. Anupama Kannan to do the further proceedings. Dr. Anupama Kannan.
Good afternoon, friends. At the outset, I would like to thank Dr. Desai for giving me this opportunity to introduce our next speaker, the legendary Dr. Harish Shetty. Dr. Harish Shetty, a psychiatrist par excellence, needs absolutely no introduction. In fact, his resume runs into several pages, and I was really hard-pressed to pick from his many, many achievements. However, seeing that we are in the midst of a pandemic, it would be appropriate to highlight that Dr. Shetty has worked in the mental health space after many disasters. To name a few, the Latur earthquake, the Kandla cyclone, the Gujarat Jabalpur earthquake, and the Gujarat riots. He has also provided mental health intervention to seafarers after their release from the clutches of Somalian pirates. He is a governing body member of the National Testing Agency appointed by the Government of India. Dr. Harish Shetty will today address us on the very large elephant in our room, social media addiction in COVID times. I welcome you, Dr. Harish Shetty, and we really look forward to hearing from you. To chair this session, we have Dr. Anjali Deshpande and Dr. Shirish Umate. Dr. Anjali is a senior psychiatrist practicing for 22 years in Thane. She is one of the directors of Insight Mind Care Center at Thane. And Dr. Shirish Umate is a psychiatrist and sexologist practicing in Thane since the last 12 years. Over to you, Dr. Harish Shetty.
Sir, unmute yourself. Thank you. I mean, would the chairperson would want to say anything before I start? That would be nice. No, Shailesh, Anjali. No, sir. You can start. Okay. Thank you. Thank you, Anupama, for that flattering introduction. Thank you, Shailesh and Anjali. Always there. They always come when I am speaking, and it's so nice to see them. Beautiful colleagues, some of the best psychiatrists in the country, both Shailesh and Anjali. Anjali has a lot of experience in mental health, in the mental hospital, and Shailesh has experience all over. He's seen all over also. He writes, he talks, he laughs, and he meets. Going back to what Anand said about schools being stuck in anxiety, SOPs don't run lives. Protocols don't run lives. Writing a protocol in a closed AC room is not how schools have to be started. Schools have to be started by all the pediatric task force, each one of them, to be available to the public 2-3 hours a day so that parents, teachers, and principals can be reassured. Constant reassurance is extremely essential for the schools to start, not SOPs and protocols. That's not how life runs. The school principals, teachers, and parents should have a helpline which they can call any time of the day or night. Have you forgotten that Mumbai had 1916 in every ward for COVID beds? Why can't they do that for the schools to start? Principals, teachers, and students should have access to phones so that to bring down anxiety. Collective reassurance, tons of reassurance and protocols, both will bring down the anxiety to start schools. Protocols and SOPs cannot run lives, and that is something which is very important for the task force, for the state government to understand. Chalu kara ani asa kara, asa hot nahi ho. We are in the middle of one of the greatest pandemics human race has faced. And when I say this, I would like to start by saying social media is awesome. One of the 10 most important technological discoveries in this era is social media. And because of that, we are all here. And each one of us is addicted to it. Let me be honest, doctors, patients, children, mothers, fathers, each one is addicted to it. And sometimes the addiction is beautiful and sometimes it's harmful. Well, allow me to start by saying that doctors also have low self-esteem if they're not connected with their world, and they're more connected with the screen space all the time, watching videos, watching Begums on the Netflix, on Amazon, and destroying their sleep because the addiction is so good. Gone are the days when you tell all parents in our parenting sessions, you need to actually compete with the jingle on the TV. Those days were days when we would say that actually TV addiction was very big. Those days, TV addiction, stop TV on 1 day in a week, and all that is gone. Similarly, this will also go when something new actually comes up. So when you are meeting real people more than real, self-esteem drops. Self-esteem also drops. And when it is very low, there's a tendency to go onto the social media space. And it really is a consequence and a cause. Loneliness is a consequence and a cause. I see people who are staying alone. Doctors who are shy, introverted, who talk less, are very happy in the social media space. It's not the young doctors alone. It's not the resident doctors. Doctors in the 60s and the 70s sleep late, and they feel that this helps them to connect with the big world. What also happens is love increases in real and not real. And when doctors connect with a lot of men and women across, and the energy is spent, the emotional energy bank is spent, they're not able to love people who are actually around them. Loss of relationships. I've seen so many people complaining to me that I would want to file a divorce because my husband or my wife is on the social media space for a long time. A loss of sleep and peace is important. Now, as you see this, I would like to say that social media is all about the dopamine slave. In fact, what you do when you take cocaine and what happens to your brain when you take stuff also happens similarly when you are addicted to the social media. Now, what happens? I'll give an example. Sakali utun WhatsApp check karto hai. Oh, Sakali utun, hold the toothbrush, not the phone. Spend a little more time with your breakfast and pick up the phone at least after 1 hour of getting up. Emergencies will not matter. Nothing will matter. If you are not available, patient will go to somebody else, and that is so important. What is also important is do not keep your phone adjacent to your bed if you're working as a team. If you keep your phone outside, even as a doctor, and you have a team to take care of, say, for example, there are 5 doctors practicing together, though they are sitting in different clinics, and each one says, Monday is your day for emergency. I transfer the calls at night to your phone. You can keep your phone away from yourself 5 days in a week. And that is so important. The distance between the phone and the bed is directly proportional. It is inversely proportional to the social media addiction. Also, you need to see social media addiction is when somebody else is watching, you also feel like watching. Baiko, tu kai bakte, acha, you're watching the new videos of your ministers, then I will watch somebody else. So what is so important is you sit close to each other, but your sight is on the phone. So it's also like if somebody is drinking, you feel like drinking. Similarly, this also happens when somebody is on social media. There's a lot of justification. There's a lot of rationalization. Nothing wrong in that. We all of us are in seminars. I'm in 7 seminars today. So we are all in front of the screen. But what is important at that point of time is you need to understand what is healthy and what is not healthy. So I was talking about toothbrush or screen. 100%, my dear doctors, from today, while you're eating, kiska bhi phone aane. Kisi ka, even if it's the prime minister, the chief minister, even if it's your ex-boyfriend or girlfriend, see to it that the phone is there. It will take a long time for you to do it. And slowly, if you find that if your mindfulness improves in activities, number one, if you're mindful and able to listen and concentrate to people addressing you or speaking to you inside the house, the children are speaking to you for 5 minutes, you're completely there, you'll find that your desire to hold the phone goes down. while eating, if you use all 6 senses, 5 are the senses and 6th sense is the food, and really enjoy the food in all ways, eating slowly, your attraction to the gadgets will go down. You may not sleep with your spouse in a bedroom, but have a bedroom for all the gadgets, which means so important to say that a closed drawer, which is where the phones are not visible, is a good technology or a good technique to keep you out. Now, it is so important for us to understand that we learn from all the speakers, from all the questions asked by each one of you, and it's all through screen. So when you do that, you need a separator, a judai, and which means what? Which means after this seminar, I'll finish the seminar in the next 30 minutes. Dinkar's backdrop is still very beautiful, the old Mumbai-Pune road. And after that, I'm going to keep the phone, I'm going to have my lunch and not touch the phone till 3:55 before the next seminar. I'll keep the phone outside. So that's a separator. A separator is very important when you deal with something healthy, and probably when you deal with something very unhealthy. And the separator, for example, when you come from the clinic, don't come home straight. After your clinic is over, and after you finish seeing all the patients, all the slides in the pathology lab, or all the patients in the OPD, it's important to have a 10-minute separator, which means pick up the phone and speak to your friends, or if you're offline sometimes, on the way, meet your other colleagues and just sit down for a cup of tea or a chat, or even a 5-6 minutes chat with somebody else. The separator helps you to separate from what is healthy to what is unhealthy. But if you continue having the phone across time from your seminars to your WhatsApps, things will be very, very, very difficult. Now, addiction is all the cues. Say, for example, if you look at addiction, it is important to understand that for an alcoholic, it's not the high of alcohol which is important. It's not the high. After some time, it's not the high. It is the sight of the bar as you travel, the sight of the wine shop, the sight of the bottle, the vivid imagery of a bottle, the vivid imagery of joy and having alcohol when you're younger, and the sight of the glass, the time of the day, the taste of the glass on your lips, the taste of the alcohol inside your mouth. There are a million cues. And what we do in addiction is we kill the cues, which means what? What are the cues as far as phone is concerned? And similarly, the cues are for drugs. For a brown sugar addict, it is the paper which attracts you to take brown sugar. It is a droplet which is there which attracts you to take brown sugar. It is a time of the day which is ganti bajgi, abhi mujhe glass, abhi mujhe kash lena hai. And that is important. And that is so important. So how do you kill the cues? Well, in the cue is सुबह उठ के कोई पेशेंट का कोई SMS आया क्या, सुबह उठ के किसी के अपॉइंटमेंट के लिए कुछ आया क्या, सुबह उठ के if I get up, I would want to see whether my OT which I planned, is it on or is it not on, and so or it's a habit. It's like picking up a paper, newspaper. It's like picking up a phone. Picking up the milk bottle on the gate is like picking up a phone. Putting the kachara out in the day is like picking up a phone. So focus on the newspaper, on the kachara and other things, and kill that intense desire to hold the phone early in the morning. The sight of the phone. कल उसने मुझे बहुत अच्छा जोक भेजा और बहुत अच्छा एक रील भी भेजा, बहुत अच्छा वीडियो भेजा. परसों का सेमिनार का वीडियो अभी आने वाला है. मैं चेक करूँ क्या? काय को चेक करने का भाई, चेक करने का आवश्यकता नहीं. If you can break that habit for 1 hour after you get up, the intensity of the cues will actually go down. The second most important thing is when you drive to your clinic, do you want a Bluetooth? Many people have Bluetooth and they talk and drive. Unless you are a police commissioner, unless you are an intensivist working in a hospital where you don't have a colleague who's also an intensivist and who takes over your work after you go, nothing will change. The world will run without you. Patients will abuse you even if you are fully available. Patients will abuse you even if you are partly available. So all the cues associated with the phone is important. Now, some people ask me, I'm on Instagram, I'm on Facebook, I'm on WhatsApp, and I'm except LinkedIn, I'm all there. Now my job is different. I do not I do not watch too many jokes because I myself have a great sense of humor, and I don't want to read anything which is irrational. I use it to promote mental awareness on this beautiful mental day. I use it to share content, share knowledge across all sections. So then how do I feel after that? Initially, I used to look at how many likes were there, how many people have read. Say for example, yesterday I asked, since today was the mental health, they asked a very senior actor to like and retweet my tweets. He agreed to do it. But now what happened after some time? I stopped looking at the likes because I told myself, I told myself that what is very important for me is Nishkam karma, which means I do it and forget it. I'm not completely in that. I might still say once in a week, pick up and look how many people have read the article or seen the Instagram post or even the tweet. But the point is, at this point of time in COVID times, we have gone through a lot of churning internally. We have gone through a lot of existential churning internally. We have been close to death and close to infection. That makes us humble. That reminds us of our temporary nature of our job on the earth, and that also reminds us that we should continuously do our duty and forget what happens after that. And that attitude is so important. And sometimes that attitude also helps you to get over. Now I get calls. मैं वोडाफोन से बोल रहा हूँ, आपको फ्री पैकेज है, नेटफ्लिक्स और अमेजॉन के साथ आपका फोन का बिल के अंदर आ जाएगा, आपको कोई चिंता करने की आवश्यकता नहीं. It is all seductive marketing, whether it is a phone call from Airtel or Vodafone or a phone call from anywhere. Subscription, free subscription is like cocaine. Jio gave us free internet for 6 months. And see how children are addicted to the internet. When internet was given for 6 months, a lot of kids those days watched PUBG and played PUBG among the poor slum kids when the father had free internet was for 6 months on an Android phone. So nothing is free, and subscription is like cocaine. So how much internet do you want to watch? How much Netflix do you want to watch? Do you want to actually watch it on the screen or on the phone? It's important to understand that if you share the screens, big screen is important, not the small screen alone. Your amount of time you spend on these channels will actually go down, because on the big screen others will have control. Your wife will say, your husband will say, "Bas zalata zok." On the small screen you have no control. It's only you. And I do not know how can you see a song in a small screen. The joy of the big screen is so good. So those who would want to chase the big screen will also have little attraction for the small screen. So any subscription which you get free of cost is cocaine. Somebody was saying in my house, "Abhi apun chhod denge the Vodafone for Airtel family package." But the family package will not last for a few months. Again, you'll have to pay as much as you're paying for other packages. So when you are seduced by the phone, by the platforms, don't fall for it. It's important not to fall for free things, and limit and tell yourself that you will be in so many groups. You will not be a breaking journalist to share all the good news, all the good news which is there. And it's so important to understand that if you stay away from your subscription, your number of hours will actually go down, because subscription is like cocaine. Now, what are the emotional defense systems you want to build on the phone? And we always build emotional defense systems for everything on life. Say, for example, if you have to say, "Ki Mata, I will go for the banquet, पण मी 2 जास्त घेणार नाही, माझ्या मित्राला सांगितलंय, मी 2 घेतलं तर मला तिथून खेचून आण बाहेर. I will go to the conference, but I will go home for lunch. I will not stay there for lunch for too long, because I would like to go and spend time with the family." The doctors who drink a lot and have really built emotional defense system by saying that in the evening time when the Ganti strikes, I'll be in the gym, and in the day when I travel, I will not drive my car. I will ask my driver to drive the car. so that he locks the door on both sides and travels on a road where I cannot see anything around me. So building emotional defense systems. A lady alcoholic, actually from New Bombay, said that you help me what to do. I said, see, evening time is the time when a lot of good things and bad things happen. Murders happen in the evening, satsang happens in the evening, gym also happens in the evening, and a lot of things happen. So building emotional defense system. Asking yourself, how much is your emotional contact time with your family? Increasing the emotional contact time will make you decrease your social media addiction. And also increasing your family contact time will slowly bring down your addiction to the media. Now, don't forward things to your spouse all the time. Read this and read that. Don't forward videos. There's a limit to which you can enjoy the video. Now, some people say, sir, aisa nahi, sir. Social media mein entertainment hota hai, or ka entertainment hai. Nothing wrong. Go and have a look. But what is important is live entertainment is as important as social media entertainment. Entertain each other inside the house. That is also a very powerful emotional defense system which you want to actually enact and actually do. So my dear friends, in your own way, you can build a list of emotional defense systems, just like we have an army on the border, and we build these emotional defense systems inside borders. Now, can social media treat social media addiction? Very important question. Yes, it can treat. Well, we use a lot of Facebook posts to tell people how to stay away from social media. We share a lot of articles on other platforms to help people move away from social media. How can you blame it? Social media saved lives in COVID. It has saved so many lives in COVID. In fact, a lot of beds were found because of social media. A lot of remdesivir was found because of social media. A lot of oxygen cylinders were found because of social media. I could help my friend in Agartala to get a bed in Agartala because of social media. So let's not damn the guy. He's not Ravan. He's a good guy. He is Ram and Ravan both together. We might utilize the Ram. So social media has been very helpful in saving lives. And also, can it help you in treating addiction? Yes. How? There are a lot of apps which can limit your timing on the screen. And you can use the apps to limit your time. It will tell you how much time you have spent on social media. It will tell you where have you spent. There are a lot of apps which will help you to regulate your interest also and your appetite for the same. So social media social media can actually help you treat the addiction. A lot of young boys and girls have moved out of social media because of counseling by Dr. Shailesh Mate, Dr. Deshpande, and people like us online. So that has also helped people move away from social media. I still remember this boy was playing games for 15 hours, 3 hours in the day, 10 hours a day, and 5 hours at night. And what did he do? He didn't actually come to me. And his father said he's ready to talk to you on the phone. I did not talk to him about addiction at all. I started speaking to him about his interest. And then I found that this boy was also a gymnast and he could do 2 tumbles around. And when we entered into the phase of his interest, and I also started looking at his videos of the tumble, I was so interested. Then I found out how much he spent and how much money his parents have to really help him go ahead in the sport. He said it's very difficult, it's very expensive, and it takes time to go around, and it's really very expensive. Then I said, can we help you? And we formed a group of people that could help him. And then his entire approach towards me changed. His entire approach towards me was that the uncle is helping me, and then the rapport is really stopped. So sometimes freakish intervention also help. I'll give you 2 examples of girls. There were 2 girls, one in 12th and one in MBBS, both addicted to the media. They reluctantly came to me. They looked here, they looked there, and they stared at me, said, "Tum kaun hote humko bolne wala? Tum salah social media mein akkha din hota hai." And what did I do? I did not give them any gyan. I did not do REBT, CBT. Both the girls asked them to intern with me. Can you believe this? The 12th standard girl who interned with me now is doing her MPhil in a college in North. The biggest girl who's interning with me is slowly getting track And she said that after MBBS, she wanted to do medicine, but 2 days ago she said, I want to do second. So sometimes tangential interventions, off the hook interventions, throwing a googly can also help treat social media addiction. Similarly, among doctors, there were a bunch of doctors who were going through the Kalkuta part in the morning till evening. I forced and egged one doctor to go there along with. He said, I've never trekked. I said, there's no need to know how to trek. You are young. Your heart is very young. You have good physics. Just go and see. And when he went there and he came down, he booked himself for another trek after 2 weeks. And then he said, it's going down. Sometimes entering into it, Thursday upwas, Satya Sai Baba's upwas, Mangalwari Patya, Shaniwari Shani Deva's upwas. As a social media upwas is also very good activity to limit. And sometimes deals also work. To आता ह्या दिवशी 2 तास बंद केलं ना, मी तुला माझा FD मध्ये तुझं नाव टाकतो. Also very good. I mean, you can try different things to help people. So my dear friends, as I've said, as doctors, we all compete. We want to build a practice. We want to pay our EMIs. We want to have hospitals. We want to have good homes. We want to have good holidays. But I think going back to basics is something which is very, very important. And when I say going back to basics, would mean all the time we have seen that beautiful picture. Tumhara hai, kuch nahi hai, sab maya hai, and if you do something, don't bother about the fruits. And I would say it's time that even at the age of 20, 25, 45, you be ambitious, you be aspirational, but keep on doing your Nishkam Karma, and probably it will help you. A small mention of anti-impulsive drugs, drugs for impulsivity, drugs for anger and irritability, drugs for depression at times also works in some patients of social media. Drugs for craving also helps in doctors who are intensely involved beyond the regular hours. And that I think is very important. So sometimes psychological therapies of correcting your irrational thoughts through psychotherapy is also useful. But I see a lot of doctors moving away from social media when their spiritual engagement also increases. With this, I stop and I keep my mind open for a few questions, if allowed by Dr. Dinkar. Dr. Dinkar, please. Thank you. I hand over my session to Dr. Shailesh and Dr. Anjali.
Thank you so much, sir, for wonderful presentation. As always, we have learned something. And of course, many doctors.
अजिबात नाही खुर्चीला धक्का मारायचा नाही. मी जेव्हा मायक्रोस्कोप वरती असते डोळे फुटतील माझे. जरा तरी डोकं वापरा ना. काय पाहिजे सांग मला, मी सरकते. घे ना, पण सांगशील की नाही.
आता आम्ही डोक्याचे डॉक्टर थांबा वापरूया. Very wonderful presentation, sir, as always. And from our psychiatrist fraternity, I wish all of the doctors who are present here happy World Mental Health Day. Today, coincidentally, it is there. And as you very rightly said, with some or other reasons, we indulge into social media, but we forget the purpose why we are using the social media. As you had given your purpose, that you are promoting mental health, that is a purpose. So if you are using the social media, you attach it to a purpose and leave it so that it will become a Nishkam Karma. I hand over to Anjali if she has to say a few words.
No, as usual, I think Sir's lecture was apt and perfect, and it gave a lot of insights about how social media can be good and bad. And Sir, as usual, sir, thank you, sir, for your enlightening lecture. Thank you, sir.
Back to Dinkar Desai, sir.
Hello.
Yes, sir. You are audible.
पीयूष मेरे को गायब करता है. हाँ, चलो ठीक है. So, Dr. Anupama.
Monday.
So thank you very much, Dr. Hari Shetty, Dr. Shailesh Umate, and Dr. Anjali Deshpande for giving the lecture and sharing the sessions respectively. Thanks, Dr. Anupama Kannan. I request Dr. Shraddha Seth to do the further proceedings. Dr. Shraddha Seth.
Good afternoon, everyone. Thank you, Dr. Dinkar Desai, sir. And thank you so much, Dr. Hari Shetty, sir, for an interesting lecture. So now, as we move towards our next session with very important topic in today's scenario, that is long COVID and lungs by Dr. Mehul Thakkar, I would like to introduce our chairpersons, Dr. Samir and Dr. Kinjal Modi, sir. Dr. Samir Chandatre is an eminent chest physician from Nasik and Joint Secretary of IMA Maharashtra. He is also President of International Human Rights Organization, Nasik. Welcome, Dr. Samir Chandatre. Our second chairperson, Dr. Kinjal Modi, sir, is consultant chest physician practicing since more than a decade and a half. He is fellow of American College of Chest Physician and European Diplomat of Adult Respiratory Medicine from European Respiratory Society. Practicing in PD Hinduja Khar as well as so many centers in Malad, Borivali, Kandivali area. Apart from his academics, he is a marathon runner, writes poems and presently pursuing LLB. Welcome, sir. Our today's speaker, Dr. Mehul Thakkar, is a renowned pulmonologist in Jupiter Hospital, Thane, and he has been at the forefront of treatment of critically ill COVID patients. Also, he has been participating in multiple training programs regarding pandemic for doctors specially. Welcome, Dr. Mehul, sir. Over to you, sir.
Thank you for the warm welcome. And at the outset, I would like to thank the organizers for giving me the opportunity to speak. I'll just share my screen. I hope my screen is visible.
Yes, sir.
Yeah. So let's start. Let's start for the topic for the today. That is long COVID. So as said by John Ashton, he actually wrote a paper on long COVID and said, what doesn't kill you may not make you stronger. This applies to long COVID and lungs. So we all know what are the respiratory features of COVID. We know that's fever, cough, fatigue, breathlessness, ground glass opacities on X-ray. And we see patients who come and get intubated within hours of admissions, hospital admission, get better and become normal at around 3 to 4 months with a normal chest X-ray, and most of the patients do well. But there is a subset of patients, we like to call it as long COVID. It's an uncharted area, and believe me, there's a lot of research going on in that. So this is a very recent article, October, about the long-term consequences of COVID, and it was trying to throw a light on what we are bound to expect now. So they expect a lot of continuous influx of patients with long COVID. They believe 1 in 5 patients have persistent symptoms after 5 weeks. In fact, in an Italian study, they found that only 13% were completely free of all symptoms, and one or the other symptom persisted. 2/5 of patients reported a worse quality of life, and dyspnea and fatigue remain one of the major symptoms which remained in according to some data on 60% of the patients. So what is long COVID? How do we define it? Now, this is a year ago in September 2020. They just said that not recovering for several weeks or months following start of symptoms that was suggestive of COVID, whether you were tested or not. That is called as long COVID. This was in one of the BMJ webinars. And now a year later, we have a current definition of long COVID, which should be used for defining the presence of any post-COVID symptom. And it will consist of 2 stages: post-acute sequelae, that is from week 5 to week 12, from the onset of patient symptom initially, and a chronic post-COVID symptom, which lasts for more than 12 weeks. So these are the commonest symptoms which patients experience, and most common is going to be
काय केलीस ते?
dyspnea,
हा?
fatigue, post
किती सॅम्पल
exertional malaise.
सापडली त्याची?
And
मग 2 कसं आलं आणि दोन्हीचं वेगवेगळं कसं आलं?
can I request others to yeah. So post exertional malaise and brain fog. Now there was a lot of hue and cry over brain fog, and I think an addiction to social media plays a role. So Dr. Shetty was talking about that. And when you're examining a patient, you have to see a patient as a whole. So look for all systems involvement. You can get a cardiovascular system involvement, pulmonary, renal, rheumatic, neurologic, and other symptoms of weight loss and allergies and mast cell activation syndromes. So current data suggests that long-term respiratory symptoms, these are, I'm giving all data because this is what we have currently. Nobody actually knows what is the exact symptom, which patients get it more frequently, and what is going to be the treatment. So currently 21% in one of the studies was found to be symptomatic at 5 weeks. And in a data of 3,700 patients, at almost 40% had breathlessness at the end of 7 months, which is a very significant number. Now, this is the most recent study published in September 2021, which looked at long-term symptoms and morphological and neurological correlates. It showed that COVID symptoms, dyspnea and cough, remain in more than 20% of the cases, and even lung fibrosis persist. Among the other studies, some have reported a higher incidence of pulmonary dysfunction, up from 28 to 53%. And in a British cohort, they found patient got re-hospitalized with a new or a worsened respiratory distress syndrome. And this is the PFT and CT abnormalities.
अरे तो लिंगे फोन घेत नाही, त्याचा फोन लागतच नाही.
We could see PFT and CT abnormalities. This is the data from the reference. And many of these studies have reported moderate to severe involvement, even though the CT findings may not correlate. And the latest study is from China, where 75% of severe patients landed with PFT abnormalities in future. We all know that there is a lot of clotting going on. We know that there are increased coagulopathy, arterial and venous thrombi, which can be in severe or non-severe disease. And there are a lot of microthrombi which are present, and a CTPA may miss. So if a patient of long COVID is showing a persistent symptom, maybe a VQ scan may be of help. Now, this is the ongoing study. This is a recent most study in August. This is ongoing. I'm saying the data is not yet released. And the objective of this study is to find what is the lung fibrosis or the so-called ILD picture that persists post-COVID, and establishing the novel diagnostic approaches and probably suggesting a therapeutic approach to such patients. Now, this study will publish its data subsequently, and I believe we should keep a watch for this. This is particularly, I want to show this. This is a very remarkable, a very small data presented in Radiology 2021 about 9 patients, where they did a xenon scan and they tried to see the regional RBC to tissue plasma ratios in patients whose CT scans looked normal. So they used clinical measurements of D-dimers, hemoglobin, and lung functions, which were within normal range post-COVID. Patients had symptoms and they got a xenon scans for them. So this is the data. They could see that almost most of the patients had a normal PFT, except for one here. Most of the patients are normal FVC, and even the DLCO was within the normal range. And here lies the beauty of the study. Now, this is image of a patient at 172, 6 months post-discharge, where you can see the CT scan is more or less normal. You see some scarring here, but normal. The ventilation and the RBC phases show there is a problem. This is a normal, healthy individual, and this is what is seen post-COVID. So this patient was complaining of breathlessness, and we could see a CT scan is normal, the PFT was normal, but a xenon scan showed a diffusion abnormality. So the abnormalities, the authors have observed that they reflect diffusion of xenon across the alveolar epithelium and therefore reflect the integrity of the alveolar epithelial capillary structure. This damage starts at the time of initial infections and it is persisted, and possibly alveolar capillary thrombosis along with this damage is shown in the post-mortem studies that we have seen. And these small abnormalities may persist and lead to persistence of symptoms. In such patients, the DLCO did not correlate with their ratio. DLCO was more or less normal, and maybe a xenon study may pick up patients who have early damage or who complain of breathlessness, but their CT scans and their PFTs are normal. But the only limitation of this study was it's a very small sample size of 9 patients. But this is, racking our brains like maybe, yes, we can follow this in future. Now, how do I assess a patient who comes with breathlessness and you suspect the patient has long COVID? Even though you may be a chest physician or a cardiologist or a general practitioner, use a holistic approach. Check everything from patient's physical, cognitive, psychological, psychiatric symptoms. Many patients suffer from something like a post-traumatic stress because of the isolation. So please assess those symptoms. You first confirm the diagnosis of COVID. Patient had, what was the symptoms, what was the duration, and what are the other health conditions associated. And be aware that people can have wide-ranging and fluctuating symptoms which can change in nature over time. Discuss how these things affects the patient's lives and activities. Discuss patient's experience of their symptoms, ask of their mental status, and involve a family member in post-COVID care. Now, at a primary care level, what you can do is check for asthma, check for OSAs, check for arrhythmias by doing an ECG. If you feel there is a myocardial infarction or a suspected pulmonary embolism, please refer on emergency basis to a nearby hospital. Check for fatigue, deconditioning, hyperventilation. When you're examining a patient, you have a stethoscope, you can check for clubbing, you can check for crepitations, check for murmurs, check for edema, check for saturation at rest and after walking for 6 minutes. It is said that a 6-minute walk test is the most basic and one of the easiest tests to perform in an OPD setting in a primary care. Get a chest X-ray done, which may be normal. Get an ECG done. Get a sputum done. Patient may have secondary infection. And if you feel there is any abnormality, refer to a COVID clinic or a specialty. Now, in COVID clinics, what normally we do is examine the patient, X-rays, PFTs, 6-minute walk test, CTPS or a CT pulmonary angiography, a 2D echo, if required, a cardiopulmonary exercise testing. And we try to differentiate long COVID from pulmonary embolism, pre-existing lung fibrosis, which may not have been picked up, breathing pattern disorders, or it's just deconditioning. If the investigations are normal, we usually discharge the patient, send the patient for rehab and chest physiotherapy. And many a times patients require long-term counseling. Now, since I could not find any data from the Indian guidelines, I have a Swiss recommendation for follow-up and treatment of such pulmonary long COVID syndromes. This was published recently in Respiration, very recently in 2021. So coming to the questions which you may have, should all patients of COVID have a pulmonary follow-up? Yes, definitely yes. The time frame is suggested as 3 months. Patient may be admitted under an infectious disease or a physician. That's what the Swiss recommend, that a pulmonary follow-up is required at 3 months. Should all the patients affected, including those with mild disease, have a pulmonary follow-up if they are symptomatic? Yes. Again, the time frame is 3 months, and it's a strong recommendation. Should all patients with persistent respiratory symptoms post-COVID have a pulmonary follow-up with, say, a DLCO or a blood gas analysis? They say yes, they should get it done. Should all patients with severe pulmonary COVID, as per the WHO definition, have a pulmonary follow-up and blood gas follow-up if they are asymptomatic? So for patients who have been severe COVID, a pulmonary follow-up is recommended, and please get a DLCO, a body box, as plethysmography, and a blood gas analysis, irrespective of their symptoms. Even if they say they are okay, some elderly patients may not be that mobile. Patient may just walk from the bed to the washroom, just may walk in the building, and may not have a physically active life to tell you that the patient is suffering from breathing difficulty. Should exercise testing be routinely performed? I would say it's a weak recommendation, and we don't have enough testing centers, but a simple 6-minute walk test should be performed. All my patients perform it and send me a chart if they see abnormality. Should a CT scan be routinely performed? Now, CT scan, if the patient has persistent symptoms, then yes. I showed you the xenon data of patients who are normal CT scan, but many patients will have a persistent CT abnormality, may show an ILD-like picture, may show a lung fibrosis. which can take from anywhere from 6 to 12 months to get better. And we should follow up these patients. Should cardiopulmonary exercise testing be performed? If possible, yes. If you have the facility, not many centers have it, but if you have, yes. Should patients with persistent symptoms have access to specialized multidisciplinary post-COVID clinics? Ideally, yes. Unfortunately, we don't have such multidisciplinary post-clinic well established in our Indian scenarios with the limited resources we have. But if possible, yes, or at least patients should be seen by a cardiologist, a pulmonologist, and a general physician. Now, this question: should patients with after COVID who present with new onset obstructive lung disease have empirical inhaled or a systemic steroid treatment? I believe most of us are using inhaled steroids during the hospital stay and post-discharge to control their symptoms. However, patients who present with a new obstructive disease pattern are recommended to start with inhaled steroids, and patients who have persistent cough may also benefit from that. That's the next question. Who have persistent cough, should they be given inhaled topical steroids? Yes, it helps. So we have seen that inhaled budesonide itself can help the patient get reduction in the cough. Now, should patients who present with interstitial abnormalities after evaluation of an active infection receive an empiric systemic steroid trial? Now, this is a gray area. If you can eliminate an active infection, what usually we follow as chest physicians is give a tapering dose of steroids to these patients who have a persistent symptom and persistent interstitial abnormalities. But you understand the steroid is an anti-inflammatory agent. If you see fibrotic changes, no active inflammation, giving a steroid will not benefit. You will just add to the trouble. So if you find ground glass opacities or active inflammatory changes, maybe yes, but otherwise no. Should patients after COVID who presents with sign of pulmonary fibrosis receive a specific antifibrotic drug? You will see a lot of doctors using antifibrotic drugs, but they are off-label recommendations. Currently, we don't have a clear consensus on it. We don't know whether it benefits or it doesn't benefit, but certainly giving dual antifibrotic will harm more. We have seen a lot of prescriptions with dual antifibrotics. Please don't do that. Use a specific antifibrotic, explain the patient it's off-label. I don't know whether it will work, but this can be tried. We don't have enough data, but there are studies on the way. And in next few months, we should get some recommendations for that. Now, should patients with COVID-19 who come with persistent respiratory symptoms after the acute episode undergo a rehab? Definitely, yes. A pulmonary rehab is the most important thing for a patient. Breath retraining, counseling, and spending time with the patient are the three things which cause the most impact on the patient. Your drugs are going to be always secondary. So please get the patient enrolled in a rehab program. So in summary, yes, radiology improves in most of the patients, even in severe cases, but a high number can have ongoing respiratory symptoms. If you exclude a pulmonary embolism and a pre-existing lung fibrosis, then probably you can think of adding the patient to a rehab program, check for vitamin deficiencies, check for psychological problems, refer to a psychiatrist if needed. But as far as giving physical therapy like drugs or steroids or antifibrotics, there is no much data currently available, and more research is needed. You may use something off-label, but currently more data is needed. Thank you.
Thanks, Mehul, for a superb presentation. Especially this long COVID is turning out to be a kind of syndrome with a spectrum of disease, and we need to have a proper history to identify, to suspect which actually patient falls into this long COVID kind of syndrome. And there is a lot of plethora of investigations which we can see. But I think for a country like us with resource limited settings, I think 6-minute walk test, which is a OPD kind of investigation. Most of the patients do come with a CT initially also. So with these two factors and our clinical judgment, we can advise the patient to minimum investigation and regular follow-up, which is most important, to the tune of almost 6 months. It was a very good presentation, and I think if there are any questions, Dr. Shraddha, please go ahead with the questions.
Yeah, sure. Sir. Thank you, Dr. Mehul sir, for informative talk. And thank you, Dr. Samir and Dr. Kinjal, for chairing the session. Thank you so much.
Thank you, ma'am.
Over to Dr. Shilpa Jain for the next session, please.
Dr. Samir.
Yes, sir.
You have got any questions?
No, sir. I think Mehul has done a very good job. I only added that in a resource limited settings, we should concentrate on 6-minute walk test, the lung scan picture and clinical signs, which Mehul has rightfully told that there is a spectrum of disease which we have to identify. It's not only the chest part, but other associated symptoms also, which contribute to the diagnosis. So I think that was the best thing.
Okay, can I intervene? I have a question in the chat box. Vaccine for pneumococcal and flu vaccine. Yes, that's a very important point. Any chronic respiratory disease, or for that matter, any chronic cardiac disease patient would be more prone for infections, and giving them flu and pneumococcal vaccinations will definitely help. As far as calling it lung fibrosis, as far as calling it lung fibrosis, it's very difficult to say it is lung fibrosis because things revert. So we likely to prefer to use the term post COVID sequelae, because that may change. And I think yes, that's a very important point for vaccination. It does help to prevent further problems for the patient.
Mehul,
yeah.
A small question. Nowadays, there is a rampant question going around third dose of vaccination for the COVID. Your say on that?
So actually, there was a good talk by Dr. Vedas who could speak on that. But I think, as far as I believe, if a person has already got COVID, I think the second dose or the one vaccine dose is going to be sufficient when the patient takes. And that is what most of the data has shown. Having an infection and then taking a vaccine gives you more protection than two vaccines together. Now, as far as the third dose is concerned, it will depend all on how the variants come up, how our country opens up. A country like India opening up and getting more infections, or the third wave so-called. As far as you ask me personally, I don't think third dose will be necessary. That's my personal belief. But yes, I could be very wrong. There could be data coming in that it is very useful. So that is my personal belief. Third dose may not be necessary.
The annual dose, what we take a flu, that may be useful, but at least 6 months later, that may not be used. That's what.
Yes, that's what. So annual dose, as many of our earlier speakers have said, it may become endemic like an H1N1 infection. So that time, if we get better vaccines, which can be given once a year, like we give for flu, probably yes, that may be useful. Probably yes, that may be useful.
Thank you.
Dr. Kinjal?
Yes, sir.
Any questions?
Yes, fine, sir. That's quite good. Very nicely presented.
May I ask one question, sir? This is Jyoti.
Please.
Hi, very nice and lucid presentation, Dr. Mehul. Just I wanted to know about flu vaccine, whether it should be continued as it is every year, and what should be the period between flu vaccine and COVID vaccine, if at all we want to get it.
So that's a nice question. Thank you for asking. The first thing I would say is that yes, flu vaccination should continue. In fact, for children where we don't have a COVID vaccine available, we are telling them at least take your flu shots regularly. That does help a lot of patients. Now, coming to the second question, for vaccination, the simple rule is any vaccine should not be taken within a gap of 15 days. You should not combine it together. That's what my personal belief is. Both the vaccines are not live vaccines. So if there's a live vaccine, you keep a gap of 1. But these vaccines are different. They are killed vaccines or a component vaccine. So if you keep a gap of 2 weeks, I think that should be sufficient.
Thank you.
Just to add one thing, what Mehul is believing perfectly okay, because guidelines also same things. DNR same things. A 15 days gap is required. So what Mehul believes is basically the guidelines follows what Mehul says. Great.
Thank you.
So for pathologists, you have made the topic very simple. And so many clinicians having a lot of great knowledge. And of course, all the celebrity chairpersons, they have made this in conclave very remembering and very great. Dr. Shilpa Jain, can you do the next thing?
Thank you, Desai sir, for spearheading such a wonderful conference.
Yes.
Thank you, sir. Thank you, sir.
Thank you so much.
Webinar. It is a great this thing for
Connect with each other.
pandemic, that we could have these things of like webinars. So I can get Samir from Nashik, Dr. Anand Kate and Dr. Vandana Kate from Nagpur. So many celebrity persons, so many dignitaries on one platform, sitting in their house. So
Our Jyoti madam too. She is the vice president current.
She is sitting in Solapur.
Yeah.
So it's
I think she's in lab right now.
Yeah,
And in labs, Samir. And in labs, Samir. Yes, you are right.
We could hear some conversation, hot conversation. Anyway, that is a part of it. So it is a boon for the COVID. कि पहले हमें लगता था कि we will not be meeting people and there will not be good see me, how face presentation is not there, we will not know the reactions. But yes, we have accepted this format and
वो कहते हैं ना सर कि दूरियां नजदीकियां बन गई.
दूरियां नजदीकियां बन गई. Right?
Okay, sir. Good day.
So thank you everybody. And Shilpa, Dr. Shilpa. डॉक्टर शिल्पा जैन अभी तो थी इधर. Anyway, Dr. Salil sir.
Hello.
Yeah, you are there.
Yeah, absolutely.
I welcome Dr. Salil Bendre sir. Hello, Dr. Shilpa.
So before Salil sir starts, I can you guys hear me?
Yes, Mehul, we can hear you.
Yeah, so before Salil sir starts, I may like to say something about him before the chairperson says something. Dr. Salil Bendre is an excellent...
Sir. Arey, ruko. Hello.
Yes, sir.
Dr. Shilpa is there.
Yes, sir. Okay.
Dr. Shilpa. हाँ बोलो.
Mehul, slides are seen.
Probably she is not.
Slides are seen.
Yes, we can see them. We can see them.
Okay.
Dr. Mehul, you were to say something. Please continue that.
I was going to say, apart from the academic credentials, which definitely, yes, we are going to announce, Dr. Salil sir is a multi-talented doctor. So anytime you feel that you want to learn magic, he's a magic teacher and a great stand-up comic also.
Rejoin karo फट से. Hello.
Shall I start?
Sir, just a moment.
Yeah.
The master of ceremony is there, but she has lost the contact.
Okay.
Just a few minutes. Anyway, I welcome Dr. Salil Bendre sir. He is the head of the department
Sir,
of.
am I audible now?
Aajo.
Thank you. Good afternoon, everyone. So here we come towards the end of an extremely enlightening session on COVID-19. We have with us Dr. Salil Bendre, who is going to talk about emerging therapies in COVID-19. He is HOD Pulmonology at Nanavati Hospital, Mumbai, and in charge for COVID facility. Formerly HOD Pulmonology at KJ Somaiya Medical College, and he has been conducting numerous teaching programs on various aspects of COVID. I also welcome our chairpersons, Dr. Amit Khomane, who is a consultant intensivist and physician, currently chief intensivist at Kaushalya Hospital, and Dr. Yogesh Sharma, who is head of Department of Medicine, Rajiv Gandhi Medical College, Thane. Welcome all of you.
Okay. So should I start now?
Yeah.
Okay. So good afternoon, everyone. And it's my pleasure to be associated with you. And I thank Dr. Desai, sir, Dr. Mehul Thakkar, Dr. Vedas for being instrumental in inviting me today and allowing me to share some of the slides which I have got today. You've been hearing about COVID right since morning. So you must be definitely relieved now the next half an hour and then you can rest. So I'm in between your half an hour, after which you will switch off your computers and laptops and have a good, nice nap. So I'm going to talk only for 15 to 20 minutes. So that's the good news. And another good news is I'm going to tell you something which are possibly new therapies which may come up later in COVID. So this is a diagram which I found very interesting. We have heard about COVID pneumonia, we have heard of IL-6, we have heard of tocilizumabs, and we have also heard of plasma, antibodies, so many things, even favipiravir, remdesivir. But this diagram actually explains why we are using these molecules. And it's very important for us to really understand the exact place where we use these molecules. Because for a clinician, it is not only about diagnosing and writing a prescription of take favipiravir 800 milligram twice a day and then make it 400 milligram and continue it and so on and so forth. It is also to know where exactly is this particular molecule working, how much is it working, and how effective it is. So as you can see, once we say that a person is infected with a COVID virus, that is a SARS-CoV-2, and we inhale them and they enter our pharynx, the nasopharynx, the larynx, and subsequently the lungs, we very well know it is through the ACE2 receptors. But what they do is they cause fusion. That's the first contact of the SARS-CoV-2 virus with the human body. That is, the virus connects with the mucous membrane and there is a step called as fusion. And this further goes on to initiate a response which is called as translation and proteolysis. So let's go step by step. First is that you imagine a patient who has got infected from a SARS-CoV-2 virus. A person has come to you, he says, I am getting cough, मुझे खांसी हो रही है, गले में खराश है, बुखार है, 3-4 दिन से, बहुत ठंडी लगती है, these symptoms. What has happened to him is, probably in the last 4-5 days or 7 days, these viruses entered his nostrils and they entered through the nasal mucosa, and the fusion of these virus particles happened with the mucous membranes and they led to the translation, the process of RNA translation. So we can see two molecules labeled here. One is called as ARBIDOL, Arbidol, which targets the ACE2 inhibitors, the receptors. So that is the first interaction of the SARS-CoV-2 with the human. And there is a molecule in the reckoning, in the making, which may prevent this particular fusion, or this may prevent the further cascade of reactions which happen. So remember this molecule, maybe after a year or two, you may be prescribing it also. Similar to that, there is a molecule which you have already used, which is hydroxychloroquine, HCQS. What does it do? It inhibits the viral entry and the endocytosis. So that is place where we say start HCQS, or that was what we used to be told in the guidelines also in the initial phases, where we start with HCQS. Now these are the two places where we are talking about. One is the Arbidol, then the HCQS, and there is one more drug which is mentioned up, that is the one which is written in starting from C. So you can see these three different molecules. I will go through each of them because these are the emerging therapies which will eventually come into our practice. So considering that, the next step is that these viruses now have entered the body through these receptors and led to further multiplication. The viruses have started multiplying by RNA synthesis. With the help of an enzyme called as the RNA dependent RNA polymerase. And that's where we use remdesivir and favipiravir. So you got my point. It is about HCQS. We know where it works. We've got two molecules which are the emerging therapies. They are working right in the beginning where the virus is going to yet join or combine with the mucous membranes and lead to further cascade. So there are two molecules. I'm going to tell you about them. We have already used remdesivir and favipiravir. So that is not an emerging therapy, but you should know where it is working. So it is working. So virus has entered the body. It has initiated the reaction. It has started the multiplication with the help of a RNA-dependent RNA polymerase. And that's where remdesivir and favipiravir come into picture. That's where they work. Subsequently, there is translation. That is the structural protein assembly formation. And then is the mediation and the inflammatory markers which increase, which is the IL-6. So that will be the tocilizumab, which is very, very popular. But there are other molecules also which also bind with the IL-6 receptor and prevent the IL-6 receptor activation. So this will just tell you a rough outline about what molecules we are going to get and where are they placed. I'm trying to emphasize this diagram again and again and again, because right from the first pandemic to the second wave, and now what we are talking about, the third wave, perhaps we have used many medicines. We have used steroids also, we have used remdesivir also, we have misused also, we have abused also, but we have done all this without exactly knowing where they work. So first initiation is the virus going in, binding, the membrane fusion, endocytosis. Subsequently, the translation and the multiplication, where there is the role maybe of remdesivir and favipiravir. We very well know remdesivir is not a life-saving drug. It reduces the hospital stay. And then comes what is called as the IL-6. So with this background, let me show you a few molecules which may perhaps be available to us. At the end of it, I will talk about antibodies, because that's where we are dealing with almost daily and has excellent role in preventing the progression of the SARS-CoV-2 virus to a high mortality disease. So we have got Camostat, that is how it is called or it is labeled as, and it is basically Camostat mesylate is the molecule. And as I said, since I had to cut down a lot of theory in it, I would just want to talk about the diagram which I showed you, where it primes the spike protein and prevents its further progression and pathogenesis. These are all in experimental stages, and that's why we say that they are the emerging therapies, which are the futuristic views, or possibly they are going to come sometime into our practice, or maybe they may not also, but they are definitely being studied because this has the potential to save lives, and including those belonging to the low income groups. So clinical data is being generated, human trials are expected, but this is a molecule which should be or which will be used in the very, very, very early stage of the SARS-CoV-2 infection. Okay, so it's a serine protease that primes the spike protein. This is one of the enzymes, the serine protease, and it is exactly where its entry is blocked by the Camostat mesylate, CM. So that's one of the molecules which we may see very soon, or maybe in a late times also. And as I was talking about the Arbidol, this is another molecule. So one was Camostat, second is Arbidol. It has been actually used in influenza. If you go by the history of these drugs, most of these drugs, you talk of even remdesivir, we talk of even favipiravir, these were all actually being used for influenza. They came up as a research molecule for influenza during the MERS time also, and later on we are dealing with during the SARS-CoV-2 also, parainfluenza and rhinoviruses. And what they do is, again, they stop the cell attachment. So these are the earliest place of the usage of these molecules which may see the future. Something which is going to be evident to us in the next few weeks, I should say, is molnupiravir. This, I suppose, all of us should be knowing about, because this drug will come in maybe a few weeks. 800 milligram twice a day for 5 days will be the way it will be told to be taken. And there are almost 6 to 7 pharmaceuticals, the Indian pharmas who are ready with this molecule, and it is going to be coming into your clinics very soon. It being an antiviral, obviously that it has to be used in the early time of the disease. See, whenever we say antivirals, as the cascade was shown, it has to be used either during the early phase where the virus has just entered and symptoms have started, but before the phase of inflammation, before the phase of inflammatory markers going high. So where do we put it? Say day 1 of symptoms to day 8 of symptoms. These are the best times to use the antiviral. So this particular molecule, it's very weird names. One of the talks where I was giving, one of the attendees asked me a question. Why are these molecules given such weird names? Why can't they make it very, very simple names? So there is some reason behind it, but to make it a little interesting, this particular molecule has derived its name from a Hollywood character. I don't know how many of you people have seen Avengers, but there is a character by the name of Thor, and he's got a big hammer, a sledgehammer sort of a thing. And this particular name is derived from that, as if this hammer is going to kill all the viruses by God's grace. So that is the genesis of this particular name, Molnupiravir. So it is an orally active drug, 800 milligram twice a day, 5 days. It has shown to reduce the risk of hospitalization by about 50% with the RCT being done. So it has got a very robust data which has come in now. It basically causes the virus to get confused because it works at the level of the size, the level of the nucleosides, and it causes changes in the mutations of this virus, and finally the virus stops replicating. So it's the inhibition of the SARS-CoV-2 replication, which is the way it works. So this is one drug which has got a future, and definitely in our country with 8 pharmaceuticals coming into the market, it is going to be affordable to the patients, not a very huge cost. So this may be something which we have to look forward to. There is other what we call as monoclonal antibodies, which again will come into the market. Presently, which we are using is the one which is being marketed by Cipla, which was manufactured, it's actually a molecule of Roche, but then marketed by Cipla in our country. However, we have got another molecule also, which is the Sotrovimab. It is again a monoclonal antibody, and these monoclonal antibodies are the ones which are actually game changers, I feel, in the last few months, since the time it has been launched in June, first week of June in our country, in Mumbai specifically. It has been useful in people who are at the high risk cases, as well as even among healthcare workers who have got this infection. So this particular molecule is very, very useful. And again, remember, it should be early, within the first 7 to 10 days. It has shown that there is an 85% reduction in hospitalization or deaths when compared to placebo. So this molecule, again, who should be given even 12 years of age and above, or at least 40 kg and above. It's an injectable form, monoclonal antibody, which has to be used in the early phase. Sotrovimab, that's the name. These names sound a little difficult for us to remember. As we start using it, I suppose we find it that it's going to be very often that we use it. The popular name which I think everyone talks about is the cocktail therapy. That is 2 different antibodies mixed together, and that's why we make a cocktail out of it. Similar cocktail is with Bamlanivimab and Etesevimab. See, these are 2 cocktails which are given together. Why they are 2 molecules given together? Because they bind to different but overlapping epitopes of the receptor binding domain of the spike protein. So we have to use this as a cocktail. The one which we are using is Casirivimab and Imdivimab. But please be remembering or knowing that these are definitely going to come. Sometimes it's very surprising that many health professionals yet are not recommending antibodies or monoclonal antibodies to the patients, considering their age group or diabetes or CKD or steroids. In fact, if they are used early, the risk of progression is definitely reduced. The question is about variants. There's always a query, will it work on Delta variants? So yes, in the lab environment, lab studies, it has been shown to be useful and it has been effective against the Delta variant as well. Though we have used monoclonal antibodies in our patients, and I would say almost all of them have recovered and become asymptomatic and not needed to continue steroids or remdesivir for a longer duration. So there is a definitely positive outcome of these particular molecules. One more molecule is the regdanimab, a big name again, but it's a neutralizing monoclonal antibody. It already has been granted a conditional marketing authorization in South Korea for treatment of adults with mild symptoms whose age are more than 60 years. So there are a lot of molecules in the reckoning which are going to come. As I said, the initial part, there was CAM and there was the other molecule. Then subsequently we had the molnupiravir, and now we have got these different kinds of antibodies, the neutralizing monoclonal antibodies. One more attention we need to go in for this, though it has been used by us, no doubt, baricitinib has been used, but there is more and more data now coming up that if we use it with or without remdesivir for confirmed patients, even in hospitalized children, then the requirement would be, rather, I would say there is a significant improvement. So these JAK inhibitors or the Janus kinase inhibitors have shown to be of significant use. It's again an oral drug. Our rheumatology colleagues have been using this for much more longer time than we are using it. So they have got a huge number of years of experience. But if you combine it at the correct time, and it can prevent the cyclic cascade of the particular inflammation, this would be a good molecule to be using somewhere in the moderate diseases and to prevent their further worsening and deterioration. But of all these things, and I'm sure that all of you doctors have treated COVID, what finally is the bottom line is the timing of the molecules. See, we have the knowledge, we have the medications, we know about the disease also, but either there is a delay in the patient reaching the hospital or there is a delay in the diagnosis. And if it's wrongly timed, then the product fails or the molecule doesn't give the effect the way it should be. So other Janus kinase inhibitors have not got approval. So please remember that it is baricitinib which is approved. We are using tofacitinib, but that is only if there is unavailability of baricitinib. Okay, so it has not got a primary approval. What if we are using a Janus kinase, which you should be using in the phase where the patient is admitted, needs oxygen, then please use it in the early phase. But tofacitinib is recommended only as an alternative to baricitinib. And again, one last line is very important here is patients who receive baricitinib or tofacitinib should not be receiving an IL-6 inhibitor simultaneously because they have got a different mechanism of action, different place of treatment. When we say IL-6 inhibition means already the time of baricitinib has elapsed. You can't go back to the time and start using it now and along with that give a tocilizumab. So even today we are seeing critical case patients. It's not that we have not seen them now and they're all mild cases, not so. In fact, today itself, I had to visit the COVID ICUs and I saw about 7-8 patients who are in the ICU, who are critical, on ventilator or going to be ventilated. So we still are facing issues. We still are facing problems with the treatment, but now we are a little wiser and perhaps we are more organized to know when to use which medication. Last 2-3 slides, then I'll end my talk, is the mesenchymal stem cells. They are still investigation. This comes up as a question asked by relatives who hope and have faith and hope that something works. Will stem cells work? Will mesenchymal stem cells work? Internet is suggesting of this. I read something on WhatsApp. So it's very clear they are not yet approved for this indication. And the NIH, it basically is recommending against the use of stem cells at this moment. Evidence is limited. We never know. There may be a breakthrough and we may get a good amount of treatment therapies once we are sure about the outcomes and the studies. So evidence is limited to this. Similar is the case with the GM-CSF inhibitors, the granulocyte macrophage inhibitors. These agents are again currently investigational. These are rather rather than giving a talk on emerging therapies, I have been bombarded with questions by patients and friends and relatives who have been reading and who have been trying to get more and more information about what can be done if my patient is not getting weaned off the oxygen, if my patient is worsening in spite of giving remdesivir or steroids. Do this work? Does that work? And that's where we are still, there is a definite use of these inhibitors. They have shown to be a 23% reduction in the risk of mortality, but they have still under the investigational phase and not come as a recommendation. The names are in the slides, but I don't think it's really important at this point of time. Now coming to last slide about what we are using and what is the present order is about the combination therapy of casirivimab and imdevimab. Many of the COVID doctors who are using them have found to be successful and useful. Again, as I said, it is a timing which matters. If the patient is in the 11th day or 12th day of the disease, he is requiring 4 liters of oxygen, he is breathless, he is tachypnic, that is not the time to say that we should try out a monoclonal antibody. Please, that's not the time. So monoclonal antibodies are, as the word says, antibodies. So the virus enters, stop them with antibodies. So that's the early phase of the viral disease. Who should be given? I would say it should be given, of course, with the risk of mortality being high in all the comorbid groups, in all the high risk groups, even patients with protein energy malnutrition, anemia, healthcare are one group where my opinion is it should be recommended, because we never know what kind of a virus we are inhaling and what variant it is. And it has shown to be having effect on the variants as well. So we are doing a daycare treatment of it. However, definitely IV infusion in 50 ml of normal saline over 30 minutes to 40 minutes is what is the best way. And if not admission, or then you can do on a OPD basis, though we have not done too many on OPD basis as a subcute, but 4 subcutaneous injection, 2.5 ml at different sites is what is recommended. Now, there are a lot of recommendations or indications, I should say, but I think we are all wise enough to know whom to give and whom not to give. But it is most important to know that do not start using it in people who are in the late phase of the disease, who are requiring BiPAP or NIV. One more corollary is that people who are having antibodies which are less or absent antibody levels to COVID are those who have shown to be having better response to the monoclonal antibody therapies. So I think with that, I have tried to explain that these are the possible avenues which we have, which will be coming. The latest which we'll be having is the molnupiravir. I am yet not very fluent with using it as a name so often, but probably we'll have a good trade name coming up from the pharmaceutical industry. So that's where we will end at. And the final will be the different not to be given monoclonals, those for hospitalized who require high oxygen therapies, patients who are, remember that COPD patients who are on oxygen therapy at home, but they get a COVID and their oxygen requirement increases. So that is something which we need to remember. It's not always that patient is on oxygen means don't give monoclonal antibody, because those they can decompensate very fast. And we may have a role to play. We have a role to use monoclonal antibodies in those cases as well. So with that, I end my talk over here. And I thank the audience, the organizers, Dr. Desai, Dr. Mehul, and all the senior delegates, my colleagues, friends, for sparing time for today's discussion. Thank you very much.
Yeah.
Any question?
Yeah, am I audible?
Yeah.
Yes, sir.
Yeah, this was a very excellent talk by Dr. Bendre. Very short, very to the point, and more informative. By his talk, we can say the monoclonal antibodies holds a future in further treatment in this particular SARS-CoV epidemic. Now, a word of caution has to be there. This availability of the monoclonal antibodies has to be at the institutional level. What we had seen during the epidemic, the patients carrying remdesivir with them and entering the hospital for admission. That stage should not come, because a lot of patients, particularly those who are net users and all, the first question they ask is, okay, my RT-PCR is positive. When are you going to give me monoclonal antibodies? So days are not far off where it is holding a very good promise, but we have to be using these particular monoclonal antibodies with cautions. Excellent talk, very specific. Thank you, Dr. Bendre.
Thank you so much, sir. Thank you.
Yes, Dr. Amit. Dr. Amit Khomane.
Yeah. Yeah. Thank you. Thank you, sir, for the excellent talk. Can I make just two observations as per our experience at Kaushalya? Yeah. So recently, sir, I got an opportunity to use molnupiravir for 15 of my patients. This was outside the trial, but on compassionate grounds, we got it from the government of Maharashtra. 15 patients. They were early. In fact, even for 2 patients. Audible?
You talk further.
Am I audible?
Oh, yeah. Yeah, audible.
Yeah. In fact, for in fact, for 2 to 3 patients, we could use them even when the CRP.
You are audible. You can continue.
Yeah. Sorry. I think my
there is audio visual dissociation, but you can continue. We are able to hear you.
Yeah. So molnupiravir we got to use on 15 patients. All 15 of them we used it around day 5 or day 6 when they presented to us. Most of them had their CRPs increased. CT scans were around mild, not even moderate, and a few of them had comorbidities. Fortunately, we gave them molnupiravir. 1 or 2 of them were admitted. The others we gave them on OPD basis. Nobody required further admission. Like all 15 of them did not require further admission, and their CRPs decreased over a period of time. We did alternate day or sometimes every 3rd day CRP for them. So yes, I think as sir said, molnupiravir is the next drug. I think better than at least favipiravir, that what we can quote. And so one observation on monoclonal antibody. Yes, sir. Within 10 days, there were people who came to us and they were intubated. Like we thought that they are on the 5th or 6th day of illness, but that is always a dicey thing when the first symptoms started. So most of these patients were either intubated or were on NIV requiring high dose therapy. They were already on remdesivir and on steroids as well, the low dose 40 BD kind of dose. Such patients, what we did to 7 to 8 of these patients, we used high dose monoclonal antibody therapy. That is a full dose, twice the dose, that is 1200 milligrams. And yes, their CRPs, despite remdesivir, they were not improving. Despite steroids, they were not improving. The CRPs were climbing. And these patients definitely recovered and went home well. So we were for 2 patients, we even used monoclonal against the guidelines, that is at the 14th day. And they even happened to recover well. So what we made a protocol that patients who present to us late and directly go on oxygen, and may be vaccinated or unvaccinated, we check their antibody levels. If the spike protein antibodies, we know that there is no single cutoff for a spike protein antibody levels, but if they are hovering around 100, 200 and not in their thousands, we are usually giving them a benefit of high dose monoclonal antibody after 3rd day of remdesivir when they are not improving with remdesivir or steroids, and they are actually responding even though it is a late phase. So they're actually responding to high dose monoclonal antibodies. 2 of these patients I remember were multiple myeloma patients, despite their antibody levels were almost 3 or 4, despite being fully vaccinated. We gave them the benefit of high dose monoclonal antibody when they were on oxygen, not responding to remdesivir on 5th day. Their fever vanished, their CRP started decreasing, their oxygen came off. because beyond 5 days, I had no options. We had no options to give. We do not continue much steroids beyond 5 days. We do not continue remdesivir beyond 5 days. We gave them this benefit of high dose monoclonal antibody, which was around day 8, 10, or 12, and probably, and to our surprise, they have recovered well. All of them have recovered well. So I think in the future, we are documenting these things, and in the future, maybe there be a role. There are some papers also to support this, sir. Yeah. So your comments on this.
So yeah, so when you say high dose, means you are saying that 2 vials together, you get 10 ml, 10 ml together, yeah.
20 ml, yeah.
Yeah, question to Dr. Salil. Can I ask?
Yeah.
Yeah, question to Dr. Salil as well as Dr. Pomne.
Yes.
Now we are into the era of post-vaccination COVID surge. A lot of patients we have been getting. As we know, the antibody titers in post-vaccination may last for 6 months or 1 year, we don't know. At what levels of antibodies in the patient you will recommend monoclonal antibodies for patients in a post-vaccinated era?
No. So first, let me just about that Dr. Amit's experience. That was very interesting because I have not used a double dose of monoclonal antibodies. I mean, we have been using it 2 patients in 1 vial because the studies were suggesting that there's not much of a significant change in the outcome even if we give a double dose. But that's a really, I should say, a very positive and optimistic outcome, even if it's a limited number of patients, and that may actually lead to something different in terms of the severe group of patients. So you should share more such experiences with me also. So even we will get an idea about it. About the question about this vaccination. Now, I suppose now the studies which have come up, sir, is actually those who are antibody naive. The studies, okay, they have been showing that if their antibodies are present, they have not recommended any titer, they have not elicited any specific titer more than this or more than that we should be using. But they are very clear with it. If they are in the mild or asymptomatic phase, high risk cases, comorbidities, it should be given even if they are vaccinated.
Okay.
So if go by that line, I do not perceive that if the antibody levels are high, that means you don't give it, because we really are not very specific about all these antibody levels. We know that.
Okay. Regarding the use,
what do you feel? What's your opinion, Dr. Amit?
So for people who had already missed the bus, like they came to us in a severe acute respiratory illness, like already requiring oxygen, we checked antibody levels with them. For people who were vaccinated, we checked the spike protein. The unvaccinated people, we used to check the total antibody levels. And what we did in the spike protein levels, we do not have a real cutoff, like what we should. So we, even with some people, their antibodies, even with spike protein not responding to remdesivir and all, their antibodies level were more than 4,000. But what 40,000, in fact, 40,000, 50,000. But sometimes what we found that these candidates who were not responding to remdesivir despite vaccination, despite remdesivir, steroids and all, their antibodies levels roughly, like in my experience of 15 to 20 patients for 15 patients around which we gave high dose, hovering around less than 500. So they were not in thousands. So this is what we could just gauge that the antibodies. So I cannot cut off one level or one number figure, sir. But their antibodies were really low. In fact, one interesting case that just now, sir, in your slide, what we saw. So in such patients where antibody levels were in hundreds, we gave them high dose. And sir, again, as in your slide, GM-CSF colony stimulator inhibitor. So one of our patients was a case of NHL. He's still there in the hospital. He got COVID second time. He had COVID in the first wave. Elderly gentleman, NHL, on chemotherapy, had neutropenia around and leukopenia, WBC around 800. He was given GM-CSF colony stimulating factors, and his WBC rose to around 26,000. And then he came with fever. So initially we thought it may be a neutropenic fever, but it was COVID. And we gave him the first shot of half-dose monoclonal antibody, and then remdesivir, but not steroids because he was already immunosuppressed. And he happened to improve with monoclonal antibody only. But I think then maybe a role of probably he had received GMCSF also to might have also helped in limiting his progression. So incidentally, GMCSF you would have received, which is perceived to be maybe as one of the treatments for COVID.
Yes, absolutely.
I think.
Thank you, sir, for the wonderful presentation today. We have been all very, very happy to attend the session yesterday as well as today. Thank you, sir.
Thank you.
So, thank you, everybody. Dr. Yogesh, sir. It was
Thank you, sir, for giving me opportunity to chair the session. Thank you.
It was a wonderful opportunity to work with you when you were Dean of Global Hospital, and I was coordinator of Global Hospital.
That was an excellent team we had.
I have worked always with Amit, sir, and Dr. Salil Bendre, sir, you have given twice opportunity to listen to you. We hope that COVID should not be there, but we should be able to get opportunity to listen to you for the third successive time.
Yeah,
probably we may be celebrating that time in a sort of conference.
Yes, definitely, sir.
Celebrity dignity there. Now we have got Dr. Rajan Sancheti. He is a.
Thank you.
He is a treasurer of IMA Pune, and he is very helpful to us for organizing these webinars on COVID updates. So thank you, Dr.
Thank you, sir.
Yeah. He is actually looking after Sancheti Hospital. So we are going to wind up this session, and before I hand over to do the valedictory things for association by Dr. Santosh Khaina, our Joint Secretary, I would like to make a very good announcement for our non-members who are likely to be members. Dr. Jitendra Suru, he has joined from Pune, and we welcome him in TPA. A very good announcement for those who have paid as non-members and joined these webinars yesterday and today by paying either 600 or 1200. We gave them a 2 days period to add to their figures. If they have paid 600, they can add 2400 and be the member of TPA. If they have paid 1200, then they can pay 1800 and they can become member of TPA. So we are going to give definitely in due course of time 10 CMEs minimum, 10 webinars minimum. November CME of 2 credit points is already planned. All the speakers are ready, and once this sheet is submitted to MMC, we will be submitting our 2 credit point November CME program very soon. So be assured that you will be taken care of in TPA. So those who have attended this CME as non-member, we hope that they will convert their non-membership into membership. So we will be eager to welcome them. Yes, our website is under construction, and it will be launched soon. A payment gateway also is under construction, and that will be also launched soon, so that it will be very easy. A very good news is that this program was live on YouTube, and those who feel that they have missed something in this webinar, they can go to the YouTube and they can listen to the particular lecture and they can relive the moments. So with these words, I thank each and everybody and hand over to Dr. Santosh Khaina. Dr. Santosh, please. Dr. Santosh, please unmute.
Thank you, sir. Thank you for giving me the opportunity. In this way, we have reached to the concluding part of our webinar. I'm sure everyone must have enjoyed it. Taking this opportunity, I would like to thank the TPA organizing committee for conducting the two-day webinar on such much needed topic of COVID. It was really the enlightening and the refreshing our knowledge about the COVID. First of all, I would like to thank the faculties, experts, and the chairpersons who have delivered and shared their knowledge and the experience with us. Along with that, I would like to thank the TPA members as well as non-members who actually were the part of the webinar and attended it through the YouTube as well as the Facebook. And a special thanks to our president of TPA, Dr. Dinkar Desai sir, who led us to organize the successful webinar. Last but not the least, I would like to mention about the Mr. Piyush who have given all his support. Thank you very much. Thank you. Over to Desai sir.
You can stop the recording. Dr. Sarup? Yeah.
Thank you, sir.
Hello, may I close the webinar?
Yeah.
End, na?
That is the end. You can
Thank you.